Welcome to day 2 of our public workshop on pharmacodynamic biomarkers for biosimilar development and approval. I'm Mark McClellan. I'm the director of the Duke-Margolis Center for Health Policy. On behalf of the Duke-Margolis Center, which is co-hosting today's workshop with the FDA, we're really glad to have you with us. Yesterday, we heard from a number of presenters and speakers with some up-to-date, excellent, diverse perspectives on these issues. And we're expecting another day of lively discussion and even more emphasis on looking forward as to where we go from here. During yesterday's sessions 1 and 2, the FDA and biosimilars developers discussed options and opportunities to advance PD biomarkers, as well as challenges expected that could limit the utility and additional value of developing PD biomarkers and how to address them. Because biologic products are diverse, it's difficult to generalize certain developmental principles and expectations, but the FDA and others recognize that opportunities to develop biosimilars are increasing. Opportunities to make the development process more efficient are growing with greater experience and further development of this regulatory science. And we expect that to continue, those trends to continue as more originator biologics are approved and as the experience here in the United States and the globe continues to grow at an increasingly rapid rate. As new analytical techniques become available, especially those that help characterize pharmacodynamic effects, efficiently identifying PD biomarkers may become easier and may become a more important part of these efforts going forward, particularly for the increasingly complex and newer forms of biosimilars. Regulators are doing their own studies, adding to this evidence base, assessing existing comparability data, working with global collaborators to track the growing knowledge and experience with PD data. And doing this collaboratively will allow regulators and stakeholder partners to develop and communicate best practices and potentially guidance and even standards on PD biomarker data in cases in which that data can facilitate biosimilar development. We've seen a foundation of this already with the guidance that FDA has already released and similar activities underway at other regulatory agencies around the world. We heard yesterday how efforts to advance PD biomarkers will be iterative and benefit significantly from discussions among sponsors and regulators. And at the same time, additional translational research, additional sources of evidence will also help improve the outlook and opportunity for PD biomarkers to influence progress in this very important and rapidly developing space. So turning to our agenda for today, our sessions are going to expand on the topics from yesterday and the discussions from yesterday, focusing on the translational research and technical detail that can advance PD biomarkers. We expect to hear more about how PD biomarkers have contributed to existing product approvals, the methodologic approaches to identifying and selecting PD biomarkers for biosimilars, and how the FDA and others are thinking about expanding the application of PD biomarkers into therapeutic areas and the support steps that will be needed to help make that happen successfully. During our final session, we'll highlight regulatory perspectives from colleagues, the EMA, Health Canada, and the PMDA in Japan. And as we reflect on the entire workshop during the final session, we'll especially want to consider where there are opportunities for regulators, research scientists, sponsors, and other stakeholders to work together towards improved biosimilar development, a more efficient and predictable process. We're glad to have you joining us today for this very important second day, and a special welcome and thanks to all of our presenters and speakers. Just a few items before we get started. Next, our statement of independence. As usual, we want to emphasize that Duke-Margolis serves as a neutral third-party convener, and we welcome and seek out a range of perspectives on issues. Our academic statement of independence means that neither Duke nor the Margolis Center take partisan positions, but of course, individual members and participants in these events are free and encouraged to speak their minds and express their opinions regarding important issues. Also, a quick reminder about antitrust policy. The details are on this slide. A key point here is we want to remind all of you that we are not intending to discuss any confidential, proprietary, or competitive information today, or to take any steps that would impede competition. And just as an additional disclosure, I want you all to know that I'm an independent board member at Cigna and Johnson & Johnson. Just a few reminders about working efficiently in this virtual format today. We're looking forward to lively, engaging discussions. The all of you who are participating, we want to encourage you to contribute to the workshop by submitting questions through the Zoom Q&A function. We had a really good dialogue and a lot of not only questions posed and comments made, but responses in the Q&A dialogue yesterday. It's at the bottom of my screen. Hopefully you can find that Q&A tab on your Zoom as well. Please don't hesitate to submit questions or comments, and we'll try to address as many of them during the course of the event today as possible. During the panel discussions, we ask that the panelists unmute their microphones and turn their cameras on, both when they're making some initial comments and then when we're all together in that discussion phase of the sessions as well. Jeffrey Florian, Yao-Ming Wang, and David Strauss from FDA will be moderating each of the 3 sessions today. We are glad to have them with us too. And with that, I'd like to get started and invite Jeff Florian to open our session 3.
Thank you, Mark, for those wonderful opening remarks and kind words. I'm Jeffrey Florian. I'd like to welcome to session 3, Emerging Experiences Approaches Using PD Biomarkers in Biosimilar Development. I'm an Associate Director, Division of Applied Regulatory Science in the Office of Clinical Pharmacology at the FDA, and I'll be moderating this session. So during this first session today, we'll hear a series of presentations from different scientists focused on pharmacokinetic and pharmacodynamic information that has been used to support biosimilar evaluation from published examples. We'll also hear a presentation on biosimilar design and development with a focus on feasibility assessments for PD biomarkers applied to biosimilar development. And lastly, we'll hear about a presentation about scientific and study considerations for evaluating drug effects and pharmacodynamic metrics, specifically related to hematologic markers. I'd like to welcome our presenters, Salaheldin Hamed, the Acting Biosimilars Lead from the Division of Clinical Pharmacology 1 and 2, Office of Clinical Pharmacology, FDA, Andres Skirjanik, Head of Clinical Pharmacology, Biosimilars at Sandoz, and Wojciech Krzyzanski, Associate Professor in the Department of Pharmaceutical Sciences at the University of Buffalo. And after we hear their presentations, I'll moderate a discussion including our presenters, as well as 2 additional speakers. Patrick Archdeacon, the Medical Officer in the Office of Medical Policy within CDER at the FDA, and Hans Al, a Clinical Pharmacology Reviewer and Team Leader in the Office of Clinical Pharmacology at the FDA. Salaheldin, thank you for joining us today, and we're looking forward to hearing more about PD biomarkers as they featured in biosimilar approvals to date.
Good morning, and thank you, Jeff. It's really my pleasure to be part of this workshop and start off session 3. As Jeff mentioned, my presentation will focus on the pharmacodynamic biomarkers that have contributed to approvals of biosimilar products thus far. Next slide. So I start with the disclaimer that the opinions presented are those of the speaker and are not to be misconstrued as the FDA's recommendations or current thinking. Also, all the information that are presented in this presentation are entirely public. Next slide. So as an outline, I will start with a brief introduction that recaps some of the information presented in session 1 by Dr. Yao-Ming Wang and in session 2 by Dr. David Strauss. Specifically, I will provide an overview of the approvals based on PD biomarkers and summarize the PD marker characteristics. Then I will move to discuss 2 case examples. The first one is about G-CSF products, filgrastim, pegfilgrastim. And the second one is about Epoetin. I will finally close with some remarks and take-home messages. Next slide. So this diagram highlights the type of clinical evidence supporting the approval of biosimilar products. So typically, PK similarity is essential for approval. In the cases where there are adequate PD markers, typically a PK and a PD similarity combined in one study is required to support approval. And safety data, which includes thorough assessment of immunogenicity, are obtained from another study. In this context, the PD similarity information is sufficient to conclude similar efficacy to the innovator product. If there are no PD markers available, a comparative clinical study in one of the indications for which the reference product is approved is required to demonstrate similar safety and efficacy and support approval. And typically these studies would be longer studies and include a larger number of patients. Next slide. Uh, Basically, we have 30 biosimilar products have been approved thus far, 22 of which, or 73%, were approved based on a comparative clinical study that compared the efficacy and safety of the proposed product to the reference product. And the remaining 8 products listed on the right were approved based on PD markers. And these include filgrastim, pegfilgrastim, Epoetin, and most recently, an insulin interchangeable product. Next slide. So the characteristics of the suitable biomarkers were discussed in detail yesterday in sessions 1 and 2, but I will just summarize them quickly. Typically, the biomarker should be relevant to the mechanism of action of the product. It should be sensitive to detect differences between products. That is, it should have a wide dynamic range, and the dose that is selected should be on the steep part of the dose response curve. Also, the biomarker should have a well-characterized profile, meaning a time of onset, the maximum PD response, and then return to baseline relative to dosing. Last but not least, the PD marker should be reliably measured, that is, having adequate and validated assays that are available to measure the PD response. So in the next few slides, we will see how these aspects apply to the PD markers supporting approval for the case studies that I've chosen. Next slide. So filgrastim is a 175 amino acid protein. It is a human granulocyte colony stimulating factor. Pegfilgrastim is essentially the same molecule, but it's pegylated to increase the half-life. So the G-CSF products regulate the production of neutrophils within the bone marrow and enhance downstream function like phagocytic ability. These products also stimulate the bone marrow to produce a large number of hematopoietic and progenitor stem cells. Those who are, the cells that are CD34 positive cells, and mobilize them into the bloodstream. G-CSF products are approved for the treatment of neutropenia associated with chemotherapy treatment or myeloablative therapy or severe chronic neutropenia. The clinical endpoint supporting these indications was the decrease in the duration of severe neutropenia. And just to note, severe neutropenia is defined as the decrease in absolute neutrophil counts to 500 per microliter or less. In addition to the neutropenia indications, filgrastim is approved for the mobilization of autologous hematopoietic progenitor cells in the peripheral blood for collection by leukapheresis. So the two dynamic biomarkers that support approval for filgrastim are absolute neutrophil counts and CD34 positive cells. For pegfilgrastim products, on the other hand, they're approved based on absolute neutrophil counts. Next slide. So we see that the pharmacodynamic biomarkers are closely related to the mechanism of action and are very closely related to the clinical endpoint. The figure on the right is showing the relationship between ANC counts and filgrastim doses. Some of the features that are very evident from the pharmacodynamic profile. First, there is a wide dynamic range in the pharmacodynamic response with dose. There is also a dose-dependent increase in the ANC counts with saturation of response at higher doses. The diagram also shows that the lower dose range may be more sensitive to detect differences between products compared to the higher doses, where saturation of effect may mask differences. So the pharmacodynamic profile is also well characterized. There is an increase to maximum ANC levels within 12 hours, followed by a decrease to baseline thereafter. As such, the PD endpoint of maximum absolute neutrophil counts and area under the effect curve captured the pharmacodynamic profile of filgrastim and pegfilgrastim and were established as PD endpoints for demonstration of PD similarity. Additionally, for filgrastim products, the CD34 positive cells exhibit similar trend to those of the ANCs. Next slide. So for the approved products, the PD endpoints of maximum ANC counts and the area under the effect curve met the acceptability criteria, where the geometric mean ratio for each parameter, along with the 90% confidence interval, were between the pre-specified margin of 80 to 125%. Additionally, for filgrastim products, the same acceptability criteria were met for the CD34 positive cells. Next slide. Another product that is approved based on PD similarity is Epoetin. Epoetin is a 165 amino acid protein that is similar to endogenous erythropoietin. It stimulates the process of erythropoiesis, which is basically the production of red blood cells. It is approved for the treatment of anemia resulting from chronic kidney disease or treatment with chemotherapy. Epoetin stimulates the release of reticulocytes, which are the precursor red blood cells, and leads to increase in RBC formation, which is eventually reflected in the increase in hemoglobin levels. Again, going back to the clinical endpoint, anemia is defined based on decrease in hemoglobin levels below a certain threshold, and Epoetin was initially approved based on the clinical endpoint of decreasing the proportion of patients receiving transfusions. So the two PD markers supporting Epoetin approvals are reticulocyte counts and hemoglobin levels are very closely related to the clinical endpoint as well. Slide 11, next. Again, we see that the pharmacodynamic biomarkers are closely related to the mechanism of action and are closely related to the clinical endpoint. So the figures on the right, they're showing the relationship between maximum reticulocyte response and Epoetin doses. Again, some features are apparent from the pharmacodynamic profile, just similar to the pegfilgrastim products and the filgrastim. There is a good dynamic range in the pharmacodynamic response with dose. There is a dose-dependent increase in reticulocyte response, and the pharmacodynamic profiles are well characterized. There is an increase to maximum reticulocyte response by day 10, followed by a decrease to baseline by day 20. As such, the PD endpoint of peak reticulocytes and area under the effect curve captured the pharmacodynamic profile pretty well, just like we've seen for the pegfilgrastim product. And these features were also reflected in the hemoglobin levels. Next slide, please. So for Epoetin products, the PD endpoint of peak reticulocyte response and the area under the effect curve also met the acceptability criteria, where the geometric mean ratio of each parameter, along with the 90% confidence interval, were between the pre-specified margins of 80 to 125%. Also, the area under the effect curve for hemoglobin met the acceptability criteria. Next slide. Before I conclude, I want to share a very important point to illustrate the benefit of using PD endpoints compared to a clinical endpoint. The top diagram highlights the relationship between the pharmacodynamic endpoint, the area under the effect curve, and the clinical endpoint of duration of severe neutropenia. As you can see, there is a reverse correlation between the PD endpoint and the duration of severe neutropenia. More importantly, modeling and simulation exercise in the bottom diagram shows the magnitude of change in the PD endpoint on the x-axis relative to the magnitude of change in the clinical endpoint on the y-axis. So it is basically the relationship between the mean difference in duration of severe neutropenia on the y-axis as a function of the percent change of AUC from the mean AUC on the x-axis. The bottom figure illustrates a very important fact that up to 40% change in the pharmacodynamic marker result in a very modest change in the duration of severe neutropenia, less than 1 day. As such, the PD endpoint is a more sensitive measure of detecting differences between products compared to a clinical endpoint. And next slide. To conclude my talk, some of the the important key points that I wanted to highlight is that the PD markers that supported the approvals of biosimilar products exhibit the characteristics of a biomarker. They're relevant to the mechanism of action. They're sensitive based on a wide dynamic range and a selection of the dose on the dose response based on the dose response relationship. They have a well-characterized PD profile that includes the time of onset, the maximum response, and return to baseline. And even though we did not discuss this point today, they are measured using validated and adequate assays. We have also seen that the PD markers provide high sensitivity to detect differences between products compared to a clinical endpoint, which leads to smaller sample size studies and shortened development program durations. And the most important point that I would like to close with is that the pharmacodynamic markers that have supported approval so far are very closely related to the clinical endpoint. For example, ANCs are closely related to the definition of neutropenia. Hemoglobin levels are closely related to the definition of anemia. We are yet to see approvals based on biomarkers that do not directly correlate with the measure of clinical benefit. With that, I will conclude my talk, and I thank you so much for your attention.
Thank you for that presentation, Salahildin. And next we'll hear from Andres Skurjanek with his talk. Andres, I turn it over to you.
Thank you. Good morning and good afternoon. I very much welcome the opportunity to speak with you today about the assessment of PD biomarkers in support of immuno-oncology biosimilars development. And with that, I also hope that my presentation will help advance the dialogue further about the use of biomarkers in biosimilar development. Next slide, please. So what I'd like to discuss over the next 10 minutes is about our experience with the analysis of published data on PD biomarkers in immuno-oncology, as this is an area of great interest for the future wave of biosimilars, but it is also presenting a unique set of challenges associated with these therapies, as clinical trials can be conducted in patients only at approved treatment regimens, with challenges associated with the definition of clinical endpoints and response margins, and together with ever-evolving standard of care treatments. Therefore, the application of biomarkers to reduce the patient burden with extensive clinical experimentation is of great interest to us. And I will share with you a case study with interleukin-18 analysis, with particular emphasis on sensitivity aspects of this biomarker for clinical assessment of similarity. And then I'll conclude my presentation with a summary of key learnings and recommendations and thoughts on the future direction of the biosimilar development in immuno-oncology space. Next slide, please. So as I previously mentioned, we conducted a comprehensive literature research using the GoBiome database, which identified 95 biomarkers across a broad range of PD-1 and PD-L1 immunotherapeutics. And using biomarker mapping criteria as per the FDA guidance recommendations, we identified IL-18 as the only remaining biomarker that could be potentially used for clinical assessment of similarity. And then we took a closer look at the published data for this biomarker to conduct further feasibility assessment. Next slide, please. Data on IL-18 that we used in our feasibility assessment come from a recent publication on atezolizumab, where the originator conducted a comprehensive pilot clinical study in patients with relapsed refractory non-small cell lung cancer at multiple dose levels at 3 different body weight based doses and 1 flat dose of 1200 milligrams, which is currently one of the approved doses. And the study explored more than 90 plasma markers to inform a relationship between the biomarker response and drug exposure. The authors then developed a PK/PD model that integrates drug pharmacokinetics, IL-18 systemic time course, and tumor shrinkage. And then we adopted this model to further characterize the dose-response relationship, dynamic range of IL-18 response, and assess the sensitivity of IL-18 response in the context of similarity assessment by varying systemic drug exposure using systemic drug clearance as a surrogate measure of variation in drug characteristics. And then we also applied 2 PD metrics for these analysis. One was baseline corrected area on the effect curve derived by us through modeling of the IL-18 response time course. And second was the relative change from baseline at day 21 that was shown by the originator to be a predictor of tumor shrinkage. So using the published PK/PD model and the mentioned PD metrics, we were able to capture the main characteristics of what we consider a well-defined dose-response relationship. Next slide, please. And this is illustrated on this slide. On the left, we have represented a dose-response characteristics using area under the effect curve as a PD metric. And on the right hand side, the same is shown for relative change from baseline at day 21. Both plots provide visual guides to inform dose response and location of approved dose on the dose response curve, which is here indicated by a vertical dark line, and dynamic range of response indicated as a green vertical line, the coefficient of variation or variability of response across the entire dose range marked as red line. And the section on dose response curve highlighted in green is what we call a sensitivity region to show the extent of biomarker response as a function of incremental changes in drug exposure. Both PD metrics suggest moderate sensitivity for assessment of similarity, low dynamic range, and high variability. This is not to say that such dose response characteristics are not informative for dose selection, for example, or treatment posology during new drug development. On the contrary, they are essential, but they are of limited use when assessing small incremental changes in drug exposures, as is the case with PD similarity assessment, especially when using established equivalence statistical criteria. So to get a more quantitative assessment and how these observations translate into numerical values, we assess the sensitivity of PD responses at the approved dose of 1200 milligram, which is close to the dose response plateau. And for illustrative purposes, we also assessed the 500 milligram dose, which is in the middle of the dose response curve, or what we can consider the steep portion of the dose response curve. Next slide, please. So the results for the two doses show that for a relatively high systemic exposure variation, we varied clearance by 20%. The changes in PD response metrics for both doses remain well below the changes induced by a PK response. The sensitivity is slightly better for a 500 milligram dose located on a steep portion of dose response, although still low, and would become even lower should one vary systemic exposure by a lower degree at let's say 10% variation, which we prefer typically, as it is better aligned with the variation in systemic exposure seen between test and reference treatments in published literature, and is also better aligned with the established statistical equivalence criteria. And to put the aspect of sensitivity into a trial design considerations, the very high degree of between subject variability seen for IL-18 across a dose response continuum would call for clinical sample size approaching or even exceeding the sample size required for a comparative efficacy study. So this brings me to my last slide, where I'll summarize our learnings and outline some thoughts and recommendations. So in our view, efficacy and biomarker endpoints have limited utility for clinical similarity assessment due to low sensitivity, simply as clinically approved doses are optimized towards maximum response. And sensitivity and dynamic range of drug response are closely linked. And in our experience, response range is generally too narrow to meet the sensitivity criteria required for addressing structural variation at the molecular level and associated residual uncertainty. Regarding dynamic range and the typically narrow range, we believe this is simply due to physiological limits that are part of body's intrinsic control mechanisms. So in our view, pharmacokinetics remains the most sensitive clinical endpoint and could be readily used as a primary endpoint in the field of immuno-oncology biosimilars, together with safety and immunogenicity assessment, due to strict adherence to approved posology in patients. And with that, comparative efficacy study could be readily waived by the combination of in vitro bioassays and pharmacokinetics. Next slide, please. And I would like to acknowledge members of my clinical pharmacology team who provided expert support with the work presented in this presentation. Thank you.
Thank you very much for that presentation, Andres. It certainly echoed some of the same points that we heard during day one yesterday. So finally, for our last presentation of this session, I'd like to turn over to Wojciech Krzyzewski. Wojciech, floor is yours.
Next slide, please. So I would like to talk today about the impact of the tolerance effect of calculations of area under effect curve for hematological biomarkers. Next slide. So I will start by introducing the area under the curve metric as a measure of the net pharmacodynamic response. I will briefly talk about cellular markers for biologics affecting hematopoiesis. I will talk about tolerance phenomena in hematological responses, and then I provide some insights on calculations of AUEC for markers that exhibit tolerance. Next slide, please. So this slide is meant to explain the concept of AUEC as a marker of the net effect. It is the area under the curve that is plotted for the effect versus time. It starts at the baseline, reaches some maximum level, and then returns to the baseline. So based on that, we use that as a marker of the effect. And the feature of this curve I would like to focus on is the fact that some responses to some biosimilars might not end up with the curve returning to the baseline, but the curve might actually go below the baseline and then produce a nadir and then return to the baseline again. So the behavior of this curve might be more complex than typically anticipated. Next slide, please. So the examples I would like to provide are from the area of hematopoietic growth factors that will affect the cells that are produced by bone marrow. This is the hematopoietic cascade, and you see in yellow all the cells that are served as a marker of pharmacodynamic effects for blood cells. Basically, we have red blood cells, white blood cells, and then there are platelets. The white blood cells are pretty much concerned with the granulopoietic lineage. T helper cells, killer cells, and B cells are associated with lymphoid lineage. And the drugs that are currently in the development affect the productions of those cells, so the precursor of those cells in bone marrow, and that might be hematopoietic growth factors that I will talk about today, but also interleukin, and then there are some monoclonal antibodies that affect the production of all those cells. So some of those drugs will exhibit tolerance effect that I would like to explain in the next slide, please. So this slide is meant to explain the concept of the tolerance. Basically, tolerance, pharmacological tolerance, means that we have a constant exposure of a system to a drug, and the response to that exposure is not constant, it diminishes over time. So what you see in this slide is the pharmacokinetics of erythropoietin that was given subcutaneously to healthy volunteers over 4 weeks, 3 times a week. And on the right-hand side is percent reticulocytes. And then we might observe that the exposure of the reticulocytes to erythropoietin is pretty much the same over those 4 weeks, but the reticulocytes reach the steady state after the first week of treatment, and then instead of staying, being at that level, they keep decreasing over weeks to lower values. And this is a classic example of a tolerance. So next slide, please. So this is another data that shows the response of the reticulocytes to erythropoietin, but after the single dose. So for the single dose, we will see that was given in this case subcutaneously to healthy volunteers. What we see is the mean data for escalating doses. As you can observe, there is the baseline remained relatively flat. Those are the solid circles. But then every response to the erythropoietin ended up below the baseline around the 400 hours, and then slowly returned to the baseline. For some doses, it haven't reached the baseline even after 800 hours. So this is how the tolerance effect manifests itself for the single dose. And then the feature to remember is that the effect curve, as we interpreted it, would not be just the part of the curve that is above the baseline, but the effect still continues below the baseline. So it is a rebound that we see in this data. Next slide, please. Another data I wanted to share with you is the platelet response to romiplostim. Romiplostim is a peptide body that was designed to stimulate platelet production, pretty much following the mechanism of action of thrombopoietin, a hormone that produces platelets. And what we see are escalating doses given IV and subcutaneously. And then we measure the platelet count over 40 days. And then, as you can see, the baseline is in dashed line. And then the IV doses are the solid symbols, and open symbols are subcutaneous doses. And if you pay attention for the squares for the solid symbols, the highest curve, it goes up, reaches the peak, and then crosses the baseline about day 35, and that goes below the baseline. And for the subcutaneous doses, the highest dose, 2 milligrams per kg and 1 micrograms per kg, we also see a similar behavior. The peak is reached around the 12 days, and then about the 28 days, the platelet responses go below the baseline for those doses, and then they return to the baseline around the day 40. Next slide, please. So the mechanisms for the tolerance that I will provide to explain this phenomena are based on this precursor depletion or blockage phenomenon. In case of the examples I provided, this is the precursor depletion. What happens is that the precursor cells in the bone marrow are stimulated, so the remover from those precursors is the cause of causing the increase in the blood pool in the circulations, but then because of that stimulations, the precursors are depleted, and then when the drug is gone, then there's extra time necessary for precursors to replenish the overstimulated pool, and that causes the rebound. So this is the depletion mechanism for the tolerance effect, and the blockage is in kind of in the opposite way. When the drug possibly blocks the precursors in the bone marrow, and then that will cause the decrease of the response below the baseline. And when the drug is gone, the blockage is released, and that extra flux of cells from the precursor will cause a rebound. So that's the mechanism for tolerance that I believe is responsible for the reticulocytes and platelets rebounds that we saw. But there is another possible mechanism for other blood cell responses that is related to negative feedback. Negative feedback takes place when there is a mechanism from the body that pretty much tells the production cells in the bone marrow to counteract the output. So if the number of cells increases in the circulations, the signal is sent to decrease the input of cells, and then if the number of cells is decreasing, the signal is sent to increase the input. It's a very common mechanism, and that mechanism actually is how the oxygen levels coming from hemoglobin, regulated by hemoglobin, tells kidneys to produce more or less erythropoietin, that in turn stimulates production of red blood cells. So this type of feedback is different from the depletion of the precursors, but it also results in the rebound and tolerance effects. Next slide, please. So what I would suggest in terms of the using AUEC as a metric for the net effect in case of the drugs that exhibit this tolerance effect and the rebound would be actually area will be divided to two areas. One, it is the area between the baseline and effect curve, and that would be somehow reflective of the effect of the drug. And then the second area, that will be area between the baseline and rebound curve, that somehow be indicative of the tolerance effect. And then those two areas would somehow used together would explain the net effect of a drug. And then you see in these slides two scenarios. One is when there is a rebound after the inhibition of the response, and then another one that we saw in the reticulocytes and platelet data when there is a increase, the increase of the response followed by a negative. So it can go both ways. Next slide, please. So just to summarize my presentations, I wanted to mention that hematological cells are expected because of this depletion mechanism and to exhibit a tolerance, especially for hematopoietic growth factors. Tolerance for a single dose results in a rebound. When we consider multiple doses, we will see a slope in a steady state response. And then to detect a possible tolerance, we would have to carry on the studies not just at the point when the response reaches the baseline, but we should allow ourselves additional measurements just to make sure that that there isn't any rebound following the baseline reached by the response. And then second thing, we will have to have a very informative data about the baseline itself. It should be based not on one point, but pretty much points that are prior to the dose and then going up to the last point of the measurements in the experiments. And finally, when the baseline, when the tolerance effect is actually present in the responses, then instead of using AUEC as a marker, I would suggest that we should use those both areas as measures of the pharmacodynamic response. And with that, I would like to conclude and thank you for your attention.
Thank you very much for that wonderful presentation. It definitely highlights some considerations for analyzing PD biomarkers, particularly with regards to hematologic measures. So with that, that concludes our presentations for this session, and we're going to go ahead and move forward with the panel discussion. And with that, we'll be starting with Patrick Archdeacon and Hong Zhao. So first, I'd like to make a clarification on Patrick Archdeacon's role. So he is the Associate Director of the Division of Diabetes, Lipid Disorders, and Obesity within the Office of New Drugs in CDER at the FDA. He is also the signatory authority on the approval for Semglee, the first interchangeable biosimilar. And that's a very nice lead-in to what will be the first question for the panel directed towards him. Patrick, your review division at the FDA is responsible for insulin products. How does your division approach demonstrations of biosimilarity, and how do you see PD biomarkers being used in biosimilar development?
Thanks, Jeffrey. Appreciate the invitation to speak today. Really impressive talks. It's made me realize just how fortunate a position DDLO actually is in. As you say, we're the division where we're regulating insulin products, including for biosimilarity and interchangeability. So that's perhaps an especially straightforward case study. Hopefully it's still informative. The reason why I think we're fortunate is, so insulin's a relatively simple molecule. So after we finish our comparative analytical assessment, sorry, I tried to turn my video on. We really don't have much in the way of residual uncertainty. Also, we have over 100 years of clinical experience with insulins, so we feel we know quite a lot about them. We have a very well-established approach to evaluating PK and PD. So we have something called the euglycemic clamp study. Essentially what we do is we hold somebody at a fixed glucose level, give them insulin, and run in as much glucose as is needed to keep them at that insulin. So we have a really nice way of assessing time concentration and time action profiles. So that in that sense, we really can measure the binding and activation of insulin receptors. We have a good sense of the mechanism of action of use. All of this means that we were able to write a guidance some people may be familiar with, the clinical immunogenicity considerations for biosimilarity and interchangeable of insulin products guidance. In general, that lays out our thinking. We think that given a favorable comparative analytical assessment and giving an adequate immunogenicity assessment, really the only clinical data that we need is a demonstration of PK and PD similarity. And we have a way of doing that I just sort of laid out the CLAMP study. So that was the approach that allowed us to take this action this summer to do the first interchangeable biosimilar product assembly. And we anticipate essentially replicating a similar approach in most instances with insulin products going forward.
Thank you for that response, Patrick. Hong, would you like to make any remarks either about that question or the presentations that you saw?
I couldn't put my video on. Now it's on. It has some lag time. So yeah, actually, I have a few thoughts to share. So I think the biggest obstacles in terms of using PD marker replace of clinical trial is that limited data is available from innovator product or reference product. So biosimilar program has to start exploring potential PD markers. That's a tremendous work involved with the uncertainty on PD markers. So now we're seeing the ANC count and CD34 or hemoglobin levels as for the supporting of approval of filgrastim and pegfilgrastim as a low-hanging fruit or hemoglobin, Epoetin as a low-hanging fruit. But while we are exploring this during the review, we face the challenge, to see the most challenge is coming from predictive of clinical efficacy. So we struggled with that until we found from literature that the ANC can't associate with the duration of severe neutropenia. That's how then we established this relationship and make it successful in replace clinical trial and support the, filgrastim and pegfilgrastim approval. But nowadays, with advancement in our understanding of mechanism of action and advance in technologies, the residual uncertainty in biosimilar product is relatively reduced compared to earlier days, because we only can test what we know to test, right? So, and also with the experience in exploring biomarker to replace clinical trials, now we have clearly identified 5 characteristics of potential biomarkers, right? So in that, we clearly state the expectation of the biomarker is no longer like a predictive of clinical efficacy, is no longer a requirement, but nice to have and beneficial. So that kind of like alleviate somewhat in the criteria for the biomarker, right? So now also FDA has initiated intelligent research work to explore biomarker as presented by Jeff today. So, and also the sponsor also put their effort to explore this, right? So work together I was thinking also we should work with innovative companies, to put more effort in exploring biomarkers. Actually, this also beneficial to them to help identify right dose and optimize dose. And also, in some cases, those biomarkers can be used in to support their manufacturing chain during drug development. So with all this effort, I will see the relevant biomarkers will play more important roles in the biosimilar program. Thank you.
Thank you very much for that response. And just as a reminder to our viewers, please feel free to submit questions through Zoom. And I guess with that, I'd like to begin the discussion by this would just be a general question to the presenters and panels. What do you see as the largest obstacle to pursuing a biosimilar development program relying on PD biomarkers? I think Han touched on a few points. I don't know if there's anything to expand, but would anyone else like to chime in on this? Andres, I see your hand. Please go ahead.
Okay. I unmuted myself. Sorry. Yeah, just I'd like to touch upon a few points. The topic of lack of data and how this translates to a biosimilar developer and asking a biosimilar developer to put more effort into exploring biomarkers is in our view, really not just a tall order, because, for example, you've seen and you've done it yourself. You presented an excellent case yesterday, what it takes to do a good pilot study. And for us, the concern, well, less of a concern, but it's actually feasibility. What do you do in patient targeted therapies where you really cannot do healthy volunteer studies? And in patient studies, it's really difficult to deviate from approved doses, because really to understand properly the properties of a biomarker, you have to do a very comprehensive dose response type of a study. And that's where we see a major obstacle. It's not just immuno-oncology that I discussed today, but there are other therapeutic areas, anti-inflammatories, which can be only administered to patients where we are limited in that regards. So that is one major obstacle, in addition to the real paucity of the data that we can work with.
Thank you very much for that response. I think that's a very important point. Some populations, some drugs don't lend themselves to the traditional dose exploration that we think of with regards to pilot studies. Anyone from FDA or who would like to comment on largest obstacle they see for pursuing biosimilar development?
I'm sorry, Jeffrey, could you like to comment on which aspect of it?
Just what do you see as the largest obstacle for pursuing a biosimilar development program that will rely on PD biomarkers?
I don't know if this is an obstacle, but I guess a suggestion is thinking about the overall context of the program. I think one thing that was helpful to us was when we started to consider a little bit of, sort of taking almost a Bayesian approach or a Bayesian philosophy about pretest probability and how much added information you were getting from this PD marker. And in some cases, I think what I'm saying is this is a fit for purpose question. So I think it's important to sort of have some thoughtful exercise about what is the amount of residual uncertainty that you're trying to address, and then have a discussion about whether or not the PD or PK biomarker is adequate for that need. And I think when we started thinking about it that way with regards to insulins, our pathway became a little bit more clear.
That's very helpful. So anything you would like to add to that?
Hi, Jeff. Yeah, from the context of oncology, cancer is a multifactorial disease where you have a lot of, in one indication, you have a lot of, driver mutations and you have passenger mutations. And sometimes the biomarker may change in one direction in one disease, but it does not necessarily, go in the same direction in another disease. So that context may actually complicate the extrapolation issue and the extrapolation exercise. And then, that's one of the things that, because of the complexity of cancer, that's one of the, might be one of the hurdles that may impact the development programs using PD biomarkers.
That's very helpful. I think a lot of the comments we're hearing during this session are actually going to lead in very nicely to session 4. Maybe changing pace a little bit. We've seen a lot of analyses presented day one as well here, where different modeling and analysis approaches were used to look at data. Even some discussions from Dr. Krzansky about how one may want to evaluate data. What do you see as the role of modeling and simulation in helping to both evaluate PD biomarkers, informing pilot study designs, but then also helping to inform similarity study designs? How can we use these approaches to maybe streamline these steps or ultimately come up with simpler, easier studies? I actually will direct this towards Dr. Krzansky first, as his talk was the focus on this.
So the FDA guidelines are really comprehensive about the use this modeling and simulation as a tool to help designing the biosimilarity studies. So the one that really is in the guidelines is the helping designing the sensitivity region of the dose response curve. And Andy was presenting a very nice study where this has been achieved. And the second role is in the quantifications of the variability, and then to pretty much make some judgments regarding the power of the study and number of subjects that should be involved. So those are, from my perspective, the very important applications of the modeling simulations. But from my perspective, I'm a big fan of mechanistic modeling. I would suggest also something on in the context of understanding of the behavior and using the modeling simulations for trying to test alternative mechanism of the drug actions. If the models are mechanistic and the mechanisms are accepted by the reviewers, then we can test two type of the behavior of the responses, and then make some explanations of the variability of the data based on some competing mechanism. I have experienced that biology is complicated and might not always be just one mechanism underlying the response, but there might be competing ones, and deciding which one is dominant would be a plus. So this type of applications of modeling is not just in the area of population mixed effect modeling, but also would bring on the board the new tools that are developing machine learnings and system pharmacology modeling, which are quite comprehensive, but based on mechanism that might give some insights. So that would be my comment.
No, that's very helpful. If I could just have one quick follow-up on that. So in what you're outlining there, are you saying that there could be more focus on maybe comparing model parameters rather than some of the PD metrics that are a focus, or?
Well, this is another thing. So from my perspective, given variability of the data, I wanted to make it very strong statement. This is my personal view on this. Sometimes is what modeling allows is not just characterization of the variability, but after that we can apply this map Bayesian techniques to get individual predictions quite agreeable with individual measurements. And then as a confirmatory to our metrics like AUC based on the raw data, we can similarly calculate the AUEC for the model predicted values, and that has much less variability and behave in a kind of more deterministic way. So using that as something that would supplement our decision would be at least helpful, if not important.
No, that's very helpful. Thank you for that clarification. Would anyone from FDA or industry like to react to that? So if not, I think we can maybe go back to a point that was, Andres had touched on this during his talk. It also had come up yesterday, and I'll direct this question towards him. Could you maybe go through what are the risks and opportunities of searching for PD biomarkers for a biosimilar developer? You noted that there might be information that's not out there and that there's uncertainty around it. Could you maybe just go through those risks and opportunities again?
Sure. So, you know, as you've seen on the example that I share with you today, right? Because we wanted to focus on a newer wave of biosimilars, because we have a pretty good understanding of the cases that were discussed yesterday in terms of pegfilgrastim, in terms of insulins, also with respect to LDL lowering for anti-PCSK9, for example. So of course, one has to, when you're going into sort of a new therapeutic landscape, and as I said, one looks at all the possible opportunities, how you can inform your development strategy, development plan, and of course, biomarkers are a very attractive option. And then how one goes about this. Of course, all the data that are available are in public domain. So there is a certain degree of usable data. And then, of course, we cannot ignore what FDA recommends or what is considered a perfectly reasonable criteria for something to quantitate and put into a statistical context at the end of the day. And that's where we start, really when we start mapping those out, the biomarkers that would satisfy those criteria, we quite quickly fall short. There's very, very little left. I mean, we are aware of the markers that are out there that could fall into that category, but there are very few or almost none, for example, in the area of immuno-oncology today, right? And and when you talk about the risk, so first of all, the risk is, it's similar with pharmacokinetics. If you don't understand the pharmacokinetics well, and you go half blindly into a study, not knowing, fully understanding the disposition characteristics and the variability, you are bound to risk failing the study, no matter what. So one really, then applies different approaches, sort of, interim checks on variability. We like to do that to ensure that we are on the right track. Now, with biomarkers, yesterday we heard mention pilot studies. And again, the pilot study, a good pilot study, simply to inform the response characteristics could be relatively small, but to inform comprehensive variability behavior, it cannot be small, because you're then certainly risking a possibility that the behavior that you see on a smaller group of subjects in a clinical trial will not be replicated or seen in the pivotal trial. So, those are, having confidence in what we see, what we assess, and what we take in our development strategy is the key. And of course, then, balancing that against I would say more conventional approach. Because if it turns out that using a biomarker approach leads to excessive experimentation, large studies, then one would at the end still consider following a conventional path if possible, right? If the endpoints are well defined, if the response margins are well defined. But what I would say is, okay, I will stop with that.
No, I think that was very helpful. I mean, if I was going to restate some of the things you said, there's an opportunity cost. In short, one might be embarking on an effort to gather information, unsure of the outcome, where on the other hand, maybe there's a more definitive path. Maybe it would be longer, maybe it would be more costly, but the fact that it's set in stone and can be planned around is sometimes a more attractive option, so to speak, for developers. Would anyone from FDA like to further expand on risk and opportunities that they see in searching for PD biomarkers for biosimilars? I think Hong had mentioned a few things as she was saying. Would you like maybe to add anything to this based on the discussion you just heard from Andres?
Yeah, I've heard saying maybe just a PK is enough without a clinical trial to support a biosimilar. But I'm thinking we're not there yet because biologics are complex molecules, not like a small molecule drug. We know exactly the molecule is. So now we still need some additional supporting data to support that similarity. So that's why we're in between PK plus clinical trial and the PK/PD. So PD is a nice in between with additional to support to further reduce the residual uncertainty. I certainly see the challenges and the obstacles because we worked with biosimilar companies with a big effort to identify a biomarker, but in the end, a lot of effort put in because the nature of the biomarker, insensitive to dose response. So now we're saying you can extend larger range as long as it's safe, right? So even though it still may be more sensitive than clinical efficacy trial. So in the long run, I still think there's a PD marker exploration and the use in the biosimilar assessment is very valuable and worthwhile to pursue. And now you have FDA full support along the way. And, you can request meeting FDA and send, information request so we can address your issue. So we can stop anything that has no hope to go further, right? With that being said, I also recognize not every biologic has a value appreciate a biomarker, but we have to explore to identify to find whether it has or is not, valuable biomarker to in replace of clinical efficacy trial. That's my words. Thank you.
Very helpful. We need some hands go up. First one I saw was Patrick. Did you still have something you wanted to add to this?
Sure. I think as I recall, the specific question was whether FDA wanted to comment on the risk of doing some PD exploration. I guess the one comment I can make as a clinical review division is, I would say there's a relatively low risk of joining the BASUFA program and coming and speaking directly to the clinical review divisions. So, you may be having one set of feedback from Office of Clinical Pharmacology, and that's excellent. But we don't really get involved and give you sort of what we think would be adequate for our purposes until we meet through this BASUFA program. And I do think that was part of those types of meetings did facilitate us making decisions about what was adequate in our therapeutic area. So just thought I'd make that comment.
I definitely think it's important to emphasize the meetings with FDA through the established programs. And Andres, I see your hands up.
I would just add a part with this thought. I mean, I really, really appreciate and welcome this opportunity. I think this is a great workshop, and I think that to move forward, we need to continue talking, right? I mean, we should have more workshops like that, and also I feel that we will be more encouraged when we engage with regulators and with the FDA discussing our programs, because in our experience, the opportunities are there, but there is lots to talk, and we don't always have an opportunity to address topics in a broader sense like we are talking today, and we should be encouraged to do that when we go in front of FDA and discuss our development strategy. So I would just say let's continue talking and move this forward.
I think that's definitely a good point to bring up, and one we definitely want to echo both through the rest of the meeting and from yesterday. There's a lot of work still to be done, and that's best accomplished with open, productive conversations such as this. We have one moment. Excuse me, was there an echo? No. So I think this actually puts us at around the time for a transition. And I do want to thank everyone for the interesting discussion. Again, there are some questions in chat, and we'll look towards responding to these offline. And I'd say with that, I'd now like to turn it over to my colleague, Dr. Yaming Wang, who will be moderating session 4. Yaming, are you online?
Yes, I am.
There you are. The floor is yours.
Welcome everyone to session 4. The session is entitled Extending Biomarker Opportunities across therapeutic areas and biomarkers.
Yaming, we can't hear you.
Can you hear me now?
Yes.
Jeff, okay, great. Sorry, my audio had a glitch there. Great. Thank you, Jeff. I really enjoyed the previous session. So welcome everyone to session 4. This session is entitled Extending PD Biomarker Opportunities Across Therapeutic Areas and Advancing PD Biomarker Use in Future Biosimilar Development. I am Yoming Wang. My role at the FDA is the Associate Director for Biosimilars and Therapeutic Biologics in the Office of Clinical Pharmacology. I hope you have enjoyed the workshop. Through the presentations and panel discussions, we're getting a lot of valuable insights. We've also had a lot of very active Q&A exchanges in the Zoom. We encourage you to continue to keep them coming. So now we will have another very informative session looking into the future applications. During this session, we are going to focus on methodological advances and techniques that can advance and broaden the application of PD biomarkers into additional therapeutic areas like oncology, as we have discussed about challenges, and more broadly, like generally like what products that have complex pharmacology as well as less well characterized mechanism of actions. We will also talk about the challenges that in terms of ensuring healthcare providers are informed regarding the regulatory approach to approving biosimilars based on PD biomarkers and the benefits biosimilars can offer to patients. So we will begin with a series of presentations, and our presenters for today's session are Dr. Chen Sun, who's the biologics lead of therapeutic biologics program in the Office of Clinical Pharmacology. Dr. Paula Hyland, who is the genomics lead in the Division of Applied Regulatory Sciences in the Office of Clinical Pharmacology. And Dr. Donald Harvey, who's the professor in the Department of Hematology and Medical Oncology at Emory University School of Medicine. Our last speaker will be Dr. Bern Maibaum, who is the professor and associate dean at the University of Tennessee. After these presentations, we'll be joined by for the discussion by my colleague, Dr. Sarah Schreiber, who is the scientific reviewer at the FDA. And now I'm happy to invite my colleague, Chen Sun, to begin our session. Chen.
Yeah, thanks, Yomi. Hello, everyone. I'm Chen, the TBP biological lead from FDA. So my presentation today will focus on use of multiple biomarkers in assessing PD similarity. Next one, please. So here is my disclaimer. The presentation represents my personal views. Next one. In this presentation, we will try to address key question: when should multiple PD biomarkers be used in PD similarity study? To answer this question, first, we will see current guidance recommendation. Then we will discuss the 3 case studies to see different scenarios where multiple PD biomarkers are preferred. Finally, will be conclusion. Next one, please. So now let's see the current guidance recommendation regarding a single or multiple PD biomarkers. The 2016 FDA guidance listed here indicates biosimilar approval can rely on PK/PD approach if suitable PD biomarkers are available. Since the comparative clinical study, CCS, in patients typically takes a long time, requires larger sample size, has confounding factors. The clinical data package difference is highlighted in the bottom graph for products relying on PK/PD approach instead of PK plus the CCS approach. The PD biomarker could be a single or a composite of biomarkers that demonstrate the product's target effects. The guidance also mentions using broader panels of PD biomarkers can add additional value. In the following slides, we will see several case studies where multiple PD biomarkers are preferred. Next one, please. So slides 5 and 6 show the first case study, multiple PD biomarkers for approved filgrastim biosimilar product. The reference product is US licensed Neupogen. Neupogen is a human G-CSF. Neupogen has 2 types of indications. The first indication is for treatment of neutropenia-related diseases, such as febrile neutropenia in cancer patients. For this indication, the relevant PD biomarker is absolute neutrophil count, ANC, and it reflects mechanism of action and correlates with clinical endpoint, the duration of severe neutropenia. The second indication is to mobilize autologous progenitor cells into the peripheral blood for stem cell transplantation. For this indication, all CD34 expressed progenitor cells are important. So the relevant PD biomarker is CD34 cell count, and it also reflects mechanism of action for this indication and correlates with clinical endpoint, the required days for platelet transfusion. So the first case shows different PD biomarkers for 2 indications with different mechanism of action. Use both PD biomarkers are needed to support approval for both indications. Next one, please. So for filgrastim biosimilar product, the sponsor needs to demonstrate a PD similarity for both ANC and CD34 if they want approval for both indications. As shown in the left graph, ANC increases significantly even after a single dose of filgrastim in healthy subjects, reaches the peak concentration before 30 hours, and then returns to baseline by day 5. Based on this PD profile, the ANC PD similarity study can be designed as a single dose crossover study in healthy subjects. For CD34 cell count in the right graph, it requires multiple doses, such as 5 daily doses of filgrastim to show a significant increase. After the 5 daily doses, it returns to baseline by day 10. So CD34 PD similarity study can be designed as a multiple dose crossover study in healthy subjects. Next one, please. Slides 7 and 8 show the second case study, multiple PD biomarkers for approved Epoetin alpha biosimilar product. The reference product is US licensed Epoetin. Epoetin is a stimulating agent to increase red blood cell RBC count. Epoetin has 2 indications. The first one is for treatment of anemia caused by the following conditions. The second one is for reduction of anogenic RBC transfusion in patients undergoing surgery. Both indications have similar mechanism of action by stimulating stem cells to increase RBC count. There are 2 relevant PD biomarkers. The first one is reticulocyte count, and it reflects mechanism of action and correlates with clinical endpoints, since reticulocyte is the precursor of RBC. And in session 3, Salah has presented more details about these products. The second biomarker is hemoglobin, and it is clinically relevant endpoint, since the hemoglobin is a component of RBC. Then the question is, why are 2 PD biomarkers needed in this case? It has been reported the increase of hemoglobin is smaller in healthy subjects than in patients, since the healthy subjects have normal and a higher baseline level. However, reticulocyte count has a wider dynamic range. So adding a second PD biomarker in this case can help increase the sensitivity to detect the difference between 2 products in healthy subjects. Next one, please. As shown in the left graph, reticulocyte count increases significantly up to 100%, even after a single dose of Epoetin alpha in healthy subjects, reaches the peak concentration around day 5, and then returns to baseline by day 20. Based on this PD profile, the reticulocyte PD similarity study can be designed as a single dose crossover study in healthy subjects. For hemoglobin in the right graph, it increases slowly, only about 20% after multiple doses of Epoetin alpha for 4 weeks. So it is less sensitive in healthy subjects than reticulocyte, as mentioned before. Hemoglobin PD similarity study can be designed as multiple dose parallel study in healthy subjects. Next one, please. Slide 9 shows the third case study, multiple PD biomarkers for a biosimilar still under development, and it is a humanized monoclonal antibody. Regarding its PD biomarker strategy, first, PD biomarkers demonstrating correlation with clinical endpoints are not available. However, the comparative clinical study, CCS, in patients is too challenging to conduct. since the clinical endpoint requires non-treatment duration up to 2 years and a larger sample size over thousands of patients. To facilitate the biosimilar development, the sponsor proposes to evaluate a composite of potential PD biomarkers in healthy subjects. They conduct a pilot study, first, to evaluate a dose-response relationship for all potential PD biomarkers in healthy subjects. Secondly, to select suitable PD biomarkers based on the 5 characteristics mentioned in formal presentations. Finally, to select a sensitive dose in the steeper part of the dose response curve. So based on the pilot study, the recommended PD biomarkers in healthy subjects include the receptor occupancy to ensure similar upstream target engagement and a series of PD signals to ensure similar downstream PD responses for each indication. The third case shows evaluating a combination of upstream and downstream PD biomarkers can help reduce the residual uncertainty and accelerate biosimilar development, especially when the comparative clinical study for efficacy and safety in patients is too challenging or sometimes infeasible to conduct. Next one, please. So here is the conclusion. Under certain scenarios, as mentioned by the 3 case studies, multiple PD biomarkers may add additional values. They can address different mechanisms of actions for different indications. They can help increase sensitivity in healthy subjects, or they can help increase confidence in demonstrating biosimilarity by using a combination of biomarkers. As shown in this graph, and also mentioned by Yuming's presentation in session 1, later stage PD biomarkers reflecting clinical endpoints represent the ideal case, but are not required for PD biomarker selection, since the goal is not to reestablish the efficacy. Upstream biomarkers can also reflect mechanism of action and help accelerate biosimilar development. Next one, please. Slide 11 provides some useful resources to help you find biosimilar relevant information, FDA guidance, FDA biosimilar website, FDA approved biologics and biosimilars, including labeling and review summary, FDA purple book. Next one, please. Finally, I want to acknowledge the organization committee for this great opportunity. Also, thanks to all my colleagues listed here. The last but not the least, thanks everyone who contributes to biosimilar development and approval. That will conclude my presentation, and I will return the podium back to Yuming. Thank you.
Thank you so much, Chen. Thank you for walking through the thinking behind the use of multiple PD biomarkers in our with our examples, review examples here. Thank you. Next, we're gonna hear from Dr. Paula Hyland. So Paula, please.
Thank you, Yuming. Good morning, everyone. I'd just like to note the disclaimer at the bottom of my slide that the presentation reflects the views of the author. Next slide. So today I'm going to talk about the exploration of the utility of omics technologies for the detection of PD biomarkers for biosimilar. The outline of my talk, I'll provide a brief overview of the status of PD biomarkers for biologics and previous use of omics technologies. Also highlight some key considerations for omics-based application for the discovery of PD biomarkers and in general for biomarker discovery. And then I'm going to present some preliminary findings from our exploratory research using a specific omics technology for the identification of PD biomarkers, and then close with a summary and conclusion. Next slide, please. So since 2015, 322 biologics have been approved, including 30 biosimilars. And of the 30 approved biosimilars, only 8 included PD similarity data. And as was mentioned yesterday, many products have complex pharmacology and as a result do not have well-characterized PD biomarkers. Now, to bridge this knowledge gap, some sponsors have carried out early phase exploration using specific technologies. So further exploration of the utility of what we now consider to be progressively maturing technologies and or other technologies, as well as the analytical framework that's needed to identify relevant PD biomarkers is warranted. Next slide. So omics can be defined as the large-scale study of biomolecules and their function. And as illustrated here in this cartoon on the left, I'm just showing you what would be considered as the best known omics technologies and probably the most widely used omics technologies when we consider research in life sciences, diagnostics, and even to provide therapeutic insight. Of course, genomics focuses on DNA and DNA status, proteomics on the differences of protein levels. Transcriptomics is now looking at different RNA biotype levels, and metabolomics, of course, metabolite differences. And the latter three omics technologies, when we think about application to PD biomarkers, are probably the most relevant here because they have the advantage of capturing multiple collective pharmacological effects, which then can be used to elucidate pathways, look at network connections, and potentially inform then on the drug's mechanism of action, as well as identify potential PD biomarker candidates for further validation. Next slide. So just to highlight some key considerations for omics-based detection of PD biomarkers and any biomarker for that matter. And this involves a number of important parameters, including the study design or the experimental design, the spectrum of the groups and the treatments, even the time intervals, controlling for pre-analytical variation in your biospecimen and analytical variation. And as was mentioned yesterday, the validity metrics of the platform that you actually decide to use is very important, as well as the statistical power. And the data that is generated, it generally has to be bioinformatically processed. When we think about NGS-based data, it needs to go through a pipeline with a number of different tools. And while the tools are becoming standardized, the parameters and the thresholds can be different, which means sometimes the output can be different. And of course, this tends to be high throughput with generation of data over multiple plates, and there's a need for normalization. The statistical methods are also very important here, with a need for adjustment of correlations within and across subject variability, as well as the necessity to correct for multiple comparisons because we have multiple outcomes. And then the data itself, the signals that we see are very complex, and there's a need to understand and interpret this data and extrapolate something useful, something that can inform. And this comes, what comes into play here is a pathway-based analysis, and this is usually based on tools that use curated databases. And then last but not least, replication and validation, the necessity to technically replicate your candidate biomarker and also to independently validate it for that external validity. And that might be approached by taking a two-step discovery approach or using a discovery cohort as well as a replication cohort within your study. Next slide. So I'm just presenting two examples of omics platforms here for the detection of PD biomarkers in plasma. On the left here, we have cell-free microRNAs, which play important roles in cell communication and also the regulation of gene expression and therefore protein production. And this is an approach we are exploring in a generated and NGS-based detection method in-house, observing that the chemistry kits can produce certain biases as well as the pipeline. So what we've done is optimized what we consider to be a highly accurate, sensitive, and reproducible method that is also automated. The other method that we are looking at is the proteomics platform. And this is a method that uses a novel adapter detection system, which basically turns protein levels into measurements of single-stranded DNA. Now, proteins reflect real-time biology, and they respond to changes in intrinsic factors as well as exogenous factors, including drug treatment. And again, this method of choice is highly reproducible, has a great dynamic range, is high throughput, and allows us to look at multiple plasma proteins simultaneously in the same sample. Next slide. So to extend the proteomics technology, I'm just going to present in the next 2 slides some preliminary results from our analysis of plasma samples from the interferon beta and the PEG interferon pilot clinical studies. What's shown here is the response curve for 4 candidate proteins affected by the therapeutic dose for interferon beta over time compared to placebo. And these are ranked based on p-value and survival correction for multiple comparison. And as you can see, nice temporal patterns here with onset of response and a peak response and return to baseline. Next slide. And we see similar preliminary signals for PEG interferon, but slightly more. And again, here just showing an example of response curves for 4 candidate proteins affected by the therapeutic dose of PEG interferon over time compared to placebo. So of course, this is a work in progress for us, and it will be important to technically validate these candidates, to also look at the measure of the area under the effect curve, and as noted today and yesterday as well, to look at the variability of these potential candidates in subjects, and as well as that to independently validate these biomarkers. Next slide. So in summary and conclusion, specific omics technologies can capture multiple collective pharmacological effects. And with that comes the potential to identify novel PD biomarkers and inform on the drug's mechanism of action. A sensitive, dynamic, highly reproducible method and a well-designed discovery framework is required to achieve high sensitivity and low variability in identifying PD biomarkers using omics technologies. Our preliminary results from our proteomics platform suggest that we've identified many candidate signals that we're going to bring forward for future validation that may have potential use in PD biosimilarity studies. And future studies should evaluate other omics technologies for the identification and validation of potential PD biomarkers. And next slide. I'd just like to acknowledge the workshop organizing committee, OCP management, my director, Dr. David Strauss, the DARS Omics Working Group, and my own Genomics and Molecular Biology Group, the biosimilar guidance in the working groups, and the reviewers, and everyone who contributes to biosimilar development and approval. Thank you.
Thank you so much, Paula. It's exciting to see novel technologies being applied into this biosimilar development space. Thank you. Next, we're gonna hear from Dr. Donald Harvey. Donald, please.
Great, thank you. And I wanna thank the organizers for inviting me to speak today on clinician concerns as we think about pharmacodynamic markers in oncology, particularly biosimilar approvals. Next slide. So I think it's worthwhile to take a step back and perhaps gain the perspective of oncology clinicians and uptake of biosimilars within the space. And really the perspective of the treating oncologist or the team helping to take care of a patient when it comes to biosimilars depends on really where that patient is in therapy and the individual clinician's time and familiarity with the field of biosimilars as a whole. I find candidly many oncology clinicians know about biosimilars, but frequently don't have a very in-depth understanding of their pathway to the market and a variety of other aspects. And so I'm hoping to provide that a little bit today. But certainly within oncology care, the scenarios are different, and the scenarios are different for biosimilars or for any other aspect of therapy for where a patient is in their journey. And the first is, as many examples have already been noted, is supportive care. And so drugs like filgrastim, the G-CSF products, or the red cell stimulants, that bar is relatively low to gain clinician acceptance, because should these drugs not work or not work in a predicted fashion, generally it can be concerning, but generally it's not a very much a life or death situation in terms of the cancer therapy itself. The next setting is metastatic disease, where patients may have had a number of prior lines or are beginning therapy, and we're really hoping to try to extend life within these patients where cure is unfortunately not possible in many instances. However, we certainly want to have confidence in the therapies we're using, no matter where they're coming from. And then finally, the highest bar is curative intent, that if we are to take a reference product and replace it with something else, the clinician needs to have very high confidence that that drug is going to behave in the same way as it has for them with a reference product or other aspects. And so these three areas, I think, need to be considered as we look at the prism of the clinician in biosimilar use. And in general, I think the field continues to evolve in oncology, whereas with time, there will probably be a greater degree of confidence across all three of these areas as we use these drugs in patients and in the clinics more and more. The truisms, though, is that safety and efficacy matter the most to clinicians taking care of patients. And I'll show you some of that data that we derived from our center. And then generally, even though we talk about biosimilars not being the same as generic small molecule agents, generally clinicians think about them as generics. And I think that can be a positive or a negative depending on how your perspective is in the therapeutics field and taking care of patients with cancer. Next slide. So we actually took 77 clinicians and surveyed them here at Emory relatively early on in the biosimilar world. And we had a number of supportive care agents that have been approved, filgrastim and pegfilgrastim, and we approached a 12-question survey to these individuals, most of whom were physicians in the care of cancer patients. There were three domains and understanding of what biosimilars were and how they got to market. Prescription preferences within the given space and what they might choose should they have the ability to choose. And then finally, how much patients should be involved in the informing and selection of a drug for their treatment across the therapeutic landscape. We also performed follow-up interviews based on cost, safety and efficacy, patient preference, and disease stage in generalities, not in specifics. So next slide. Findings early on, again, were that really there was a pretty poor understanding of the regulatory process. Does a biosimilar equal generic? Are they the same thing? And 40% said yes, they are. And so, you might say, well, that's unfortunate from a regulatory perspective because certainly the pathway is different. But again, think about the end user in that if they are prescribing a drug, whether it's a generic paclitaxel or a biosimilar filgrastim, their nuanced understanding of those differences probably is not very high, and some might argue doesn't necessarily need to be that high. But again, that's probably a fairly inflammatory comment. Factors important for prescribing, there was a Likert scale with 5 being the highest. Safety and efficacy were the highest of the group, and then cost was the next highest. I point your attention to the right side where PK similarities, colleague and expert opinion, and chemical and physical characteristics were all considered less important than safety and efficacy. And while the field of biosimilars is not focused on obtaining efficacy data in a big way, certainly not in the same way as a reference product, it still matters the most to clinicians as they think about selecting an agent for treatment. In terms of shared decision-making with patients, about half thought that it was extremely important or very important, and 39% that it wasn't important. And then adjuvant use, again, moving into the clinical scenario of when a patient would get treated. If they were undecided or not confident, and 48% in adjuvant use of biosimilars, somewhat confident were 40%. So it was relatively equal in the space that would be the highest bar in general for selecting a biosimilar for care. Metastatic was a little more confident, and I think that reflects, again, early understanding of the field, and that likely, if repeated today, would probably go up substantially. So next slide. In general, now pivoting to pharmacodynamics and oncology clinicians, I like to think of my physician colleagues particularly as being relatively savvier across many areas, but I can promise you that many of them have a very limited understanding of pharmacodynamics, despite the fact that many of them use them every day and just don't call them as such. In general, it's probably limited to something along the lines of what does the drug do to the body? But it can be taught through some familiar examples like safety, neutrophil counts. surrogate efficacy. So in multiple myeloma, we use paraprotein changes to dictate how a patient is responding to treatment. And that's really a surrogate marker of plasma cell number and activity in the bone marrow compartment. There's direct efficacy of pharmacodynamics, specifically radiographic changes in tumor volumes of patients with solid malignancies. And then finally, those who have very deep involvement in translational research, things like phosphorylated protein changes in PBMCs. And that's the population of clinicians that are likely to have the greatest understanding of what pharmacodynamics can, and just as importantly, what it cannot tell us in the development of a drug or in the care of a patient. So next slide. The acceptance of PD data by clinicians, I think, is somewhat challenged because of the nomenclature and the way that it's considered in day-to-day treatment. It's not routinely used. And what I mean by that is that we don't refer to these agents and these measurements that I just suggested as pharmacodynamics. We think of them as safety measures or as efficacy measures. They don't necessarily say, hey, a neutrophil count nadir at day 8 or day 15 is a pharmacodynamic measure. They think of it as something we need to consider for taking care of patients and making sure that they're safe. When we think about no clinically meaningful difference, a phrase often cited in the space of biosimilars, that to clinicians means safety and efficacy. A clinically meaningful difference is that of a safety or an adverse event rate or an activity rate. It's not routinely thought of as PD data by clinicians taking care of folks with cancer. And the reference agents that these biosimilars are being compared to or are being benchmarked against, generally, they're not developed with PD outcomes prominently featured. And so we don't talk about receptor occupancy with monoclonal antibodies or target engagement with the direct anti-neoplastic agents that we use. We tend to not focus on that in the later stage development, and certainly not in the post-marketing space of patients or drugs that are approved in this space. And then finally, many PD markers don't directly relate to efficacy in oncology. Even for those that have receptor occupancy or other areas that are there, you may have a lot of binding. It may not mean that patients respond well because there are so many other factors that go into whether or not a patient with cancer will have a response, will have a deep response, and how long that response might last. Next slide. And so further considerations within the space of PD data and oncology clinicians is really that PD's been around for a while, and I think we have to think about it in a way that is real, and that's that we don't have a lot of uptake or expansion of pharmacodynamic data as it's currently described, certainly in research settings within that space. Secondly, that if we think about the word clinically meaningful, if we're going to have success in having clinicians adopt PD markers as surrogates, then we've got to probably change a little bit in the way we talk about it. What does clinically meaningful mean, and how can we incorporate more pharmacodynamic markers into this space? And then finally, the big question is it even necessary for clinicians to buy into the fact that a biosimilar is worthwhile for care or is truly safe and effective? Because we are truly treating them in many instances in institutions by similarly as generics. And so they're being substituted, they're being put in place of reference products across the country. And so therefore, do we really have to focus a lot on the ability of clinicians to take this data in and believe it, as opposed to saying it's gone through regulatory review, we believe in this data from a regulatory science perspective, and therefore you should have confidence that these selections have been made with informed decision-making. So with that, I think this is my last slide. I'll stop and look forward to further discussions.
Thank you so much, Donald. Your presentation gave us a lot to think about in terms of communication, in terms of collaboration with the clinicians. Thank you. Next, finally, we will hear from Dr. Bernd Maibaum. Bernd?
Well, there we go.
Great, I can see you.
Thank you, Yaming. Good morning, everyone. Thank you for the opportunity to give my perspectives here. I would like to talk a little bit about the added value of using PD biomarkers over non-discriminative clinical endpoints. Next slide, please. So you have seen this pyramid now multiple times. It symbolizes the totality of evidence established throughout a biosimilarity assessment. And as we heard this morning, as well as in the discussion yesterday, the key question remains what the clinical efficacy assessment really adds and whether it is really needed in its current form and whether it can be replaced by biomarker assessments or even completely omitted. Next slide, please. So this becomes especially relevant in the context of non-discriminative endpoints that are frequently used in pivotal phase 3 studies. So endpoints in pivotal phase 3 clinical trials in drug development for novel biologics are obviously selected, designed, and intended to show efficacy in treating a condition. And whether the outcome measures are useful in discriminating between different degrees of efficacy is at best a secondary consideration. In contrast to that, if we focus on clinical efficacy studies and biosimilarity assessments, these are intended to establish similarity in efficacy and safety between a reference product and a biosimilar. So it's a completely different question. The objective is not to establish efficacy, but to detect potential differences in the degree of efficacy. So the quantitative aspects are in the forefront. And that means we have different requirements for outcome measures. So especially aggregate clinical endpoint measures seem to oftentimes have, however, very little discriminative power, even though they are well established as clinical endpoints in many phase 3 programs. So they are usually noisy measures and they often have subjective elements. An example of that are composite outcome measures. Next slide, please. So some of these representative composite outcome measures are listed here and have been used widely, especially in the development of TNF-alpha drugs, such as infliximab, adalimumab, and etanercept, for all of which multiple biosimilars have now been approved. And I would just focus on one of them that is most frequently used, which is ACR20, so the American College of Rheumatology improvement criterion. Basically, ACR20 is a composite measure defined as both improvement of 20% in the number of tender and the number of swollen joints, and in 20% improvement in 3 of the following 5 criteria. And these are partially subjective criteria: patient global assessment, physician global assessment, functional ability measures, visual analog pain scale, and erythrocyte sedimentation rate, or C-reactive protein. So it's a relatively complex outcome criterion. And ultimately, what is then measured in clinical studies is the number of patients that actually achieve this ACR20 value, so this improvement of 20%. And you can do the same with an ACR50 and an ACR70, which are the same instruments with improvement levels defined as 50 and 70%. Next slide, please. So what you can see then here are the dose response curves on the left side for a variety of different compounds used in or used as disease modifying anti-rheumatic drugs, and included in there are adalimumab, etanercept, and infliximab that I mentioned before. As you can see, the dose response curves are relatively flat. They're shown here for ACR20 in black, for ACR50 in blue, and for ACR70 in red. What you can also see is that they look very much comparable between these different drugs. So relatively flat dose response curve. At the same time, a relatively noisy measure based on the composite nature and some of the subjective criteria included in that. Now, nevertheless, ACR20 has been used in many biosimilar approvals in the clinical efficacy studies. And one example is shown on the right side between infliximab as a reference product and SP2 as a biosimilar, given here as 3 milligram per kilogram with an induction and then a maintenance phase with 8-week dosing to over 580 patients. And you see the same kind of ACR20 time course during treatment, where the number of patients, the percentage of the number of patients treated and that receive the ACR20 criterion is basically the same between these two compounds. Next slide, please. So the value of this ACR20 assessment for biosimilarity studies is further put into question when we look at a broader range of dose response data. And these are shown here for ACR20 for infliximab from the original pivotal trials during the drug development for infliximab as the innovator. And what's shown here is a comparison between two dose levels, 3 milligram and 10 milligram per kilogram, and also in each of these dose levels, a different dosing interval, either 4 weeks or 8 weeks. And what can clearly be seen is that all treatment regimens are clearly effective compared to placebo, but no difference can be detected with the outcome parameters independent of the dose level and the dosing frequency. So everything looks the same, everything is overlapping. And that raises the question, what is really the value of clinical studies in biosimilarity assessments for TNF-alpha drugs with ACR20 as outcome parameter if the design cannot effectively discriminate between treatment effects if they would exist? So these studies surely provide safety and immunogenicity data, but one could question whether it is even ethical to perform such studies, as they would not be able to differentiate most efficacy differences if they were present. More importantly, as ACR20 as clinical outcome measure for anti-TNF-alpha biosimilars is essentially unable to detect potential differences, one could argue that the currently approved anti-TNF-alpha biosimilars are only approved based on clinical data that comprise pharmacokinetics and safety without any comparative efficacy data. Next slide, please. So what can now biomarker assessments add if non-discriminative clinical endpoints are present in a specific therapeutic area? Now, as intermediate measures of drug response, biomarkers are, of course, more directly affected by drug therapy and can be, as we discussed multiple times before, more precisely quantified. Nevertheless, of course, they can also undergo modulation, as indicated here, by a variety of different factors, as can the clinical endpoint. And the clinical endpoint may not necessarily only depend on the biomarker, but may also be affected by drug effects that are not mediated by a specific biomarker. So there's a certain uncertainty around the biomarker, how it is related to the clinical endpoint, even though it's much closer to the drug administration and the drug effect, it obviously has a certain uncertainty related to that. Next slide, please. So what are then the pros and cons for clinical endpoints and for biomarkers? Well, clearly clinical endpoints, even if they are non-discriminative, have historically been accepted in the therapeutic area as efficacy endpoint for innovator drugs. So there's no need to justify the use of an established outcome measure. And that is not only relevant for regulatory approval, but of course also relevant for the acceptance in a specific therapeutic area, similar to what Donald just outlined in the previous presentation. The downside, of course, is that the low discriminative power to differentiate outcomes between biosimilar and innovator may be present. And by that, you can wonder whether the real reason for it to potentially show lack of differences in efficacy is not really given. In contrast, biomarkers are more causally related to drug effect than variation of clinical endpoints. As we mentioned, they are more precisely measured. They usually have a larger dynamic range. And they can potentially, by that, as a more precise measure, reduce sample size requirements. The major limitation that I see is, however, that the need to justify its use, as it is not an established outcome measure in the therapeutic area. So it's not only that the assessment has to show, or that a sponsor has to show that the assessment between the innovator and the biosimilar does not show any difference. But in addition, it has to show that the biomarkers that are used for that specific purpose, if this is actually a new biomarker in a new area, is actually justified compared to the established clinical endpoint. So it's an additional hurdle that I think is largely a major hindrance why this is not embraced in a broader perspective. Next slide, please. So when you look at the cascade of intermediate intermediary biomarkers for therapeutic proteins here, then clearly biomarkers are not equal in their use and in their use to address the residual uncertainty in similarity or dissimilarity. So clearly, if you have biomarkers that are more related to target occupancy, so the target engagement, or then the pathway modulation, like target mechanism and physiological response, they are still much more related to the drug, directly affected by drug therapy. But on the other side, they are more distal to the actual efficacy outcome. In contrast to that, if you have pathway modulation and disease modulation, biomarkers for those, then you probably are much closer to the desired efficacy endpoint. So clearly the selection of biomarkers that are actually used and how many of them may need to be used is dependent on how closely they are related to one side, the drug exposure, and the other side, the efficacy. Next slide, please. So biomarkers may provide more discriminative power for biosimilarity assessments and clinical outcomes, as we mentioned. However, whether to monitor one or several biomarkers should depend on the proximity of the mechanism of action and the disease modulation. So for disease modulation, biomarkers, only one monitored PD measure may be sufficient. Whereas if you're more to the side of target engagement and pathway modulation, more than one biomarker PD measure may be needed to establish confidence in the PD biosimilarity. So biomarkers that are more discriminative and more precisely quantifiable than clinical endpoints may provide a better opportunity to identify potentially existing differences between biosimilar and innovator compounds. Of course, whether there is actually a difference, what this then means, if it's at the level of the biomarker, is obviously then a secondary question that needs further interpretation, whether it's actually of clinical relevance or not. With that, I would like to close. Thank you very much for the opportunity to share my thoughts.
Thank you so much, Bernd, for that thoughtful scientific analysis there. So with that, I'd like to thank all the speakers for the excellent presentation. And before we begin our discussion, I would like to welcome my colleague, Dr. Sarah Schreiber, to make some remarks. Sarah, are you there? Would you like to provide some remarks or react to what you have heard in this session? Any thoughts are welcome about how we can advance the use of PD biomarkers and increase the awareness of the FDA's approach to biosimilar approval. Please go ahead, Sarah. You're muted. Please unmute first. Good, it's working.
Okay, there we go. Thanks, Yami, for the introduction and participated in session 4. This is a real honor. A lot of great presentations in session 4, a lot to chew on. You know, we heard a lot about the approved biosimilar product case examples of filgrastim, pegfilgrastim, and epoetin alpha. And the work that the FDA PD research project team is doing are certainly tremendous first steps in making progress towards filling in gaps and addressing the potential for PD markers for those specific 6 products that were selected. And as Paul has just presented, the team's exploring the Omics work that's showing some very early promise. And so I think FDA will need to continue to support, have the support of all of our stakeholders in order to continue to expand upon these initial research efforts, where our goal is to really try to develop an overall evidentiary framework that can be applied broadly. However, each proposed biosimilar product is unique. And so it may be challenging to develop a broad framework until we have a diversity of more specific case examples to apply to such a framework. I've seen a lot of product-specific questions in the Zoom Q&A, which, and I don't think the focus of today is to address specific program-specific questions, but it is noted that each product is going to have its own profile and its own specifics within, and there could be variety. There's no one cookie cutter approach to biosimilar development program. Is also presented during the session by Dawn, and yesterday's highlighted by Sarah. There's a clear need for education about biosimilar products and the regulatory framework for both healthcare providers and our patients. And so FDA is certainly engaged in educational efforts to help address this issue. And once there's greater appreciation and understanding for the regulatory framework, I think the utility of well-defined and well-justified biomarkers, whether they're directly related to efficacy outcomes or if they're more distally related, I think the community should have a higher acceptance of uptake of such endpoints in the biosimilar programs. So overall, I expect this workshop to really be a kickstart to additional discussions and perhaps the formation of a multi-stakeholder working group or perhaps consortium, as Assam alluded to yesterday as well, to really continue these initial discussions around the role of PD biomarkers and expand those discussions into product-specific examples.
Thank you, Sarah. Indeed, you're very, it's well said that we do have to collaborate and partner with a lot of stakeholders to be successful in this endeavor. And we're kickstarting that whole process. Thank you, Sarah. So we're gonna start the discussion momentarily. I just wanted to have a housekeeping reminder to our viewers that you may know that there are Q&A in the Zoom, and please feel free to jot down your questions. We'll try to address them. So let's get started with the discussion. As we all heard from the presentations, that we have a lot of opportunities, very exciting with new novel technologies, and we also have challenges that has to do with many dimensions. So I would like to kind of go back to Paula's presentation, because the utility and utilization of novel technologies is sort of not new to the new drug development space and drug discovery space, but it's sort of new to the biosimilar space. So could you kind of elaborate on, this, other than this type of technology, the ones that you presented, what other technologies might be on the horizon that could be helpful, and what are there any special considerations for utilizing such technology? And after that, we'll ask other panelists to chime in with your opinion. Thank you. Paula?
Thank you, Yoming. When we were initially looking at potential platforms and technologies, again, we sort of focused on key parameters for those technologies, the validity metrics, reproducibility. So there are other potential omics technologies that could be investigated or explored. And one of them was one that I presented on the cartoon, which is, of course, metabolomics. But ultimately, it comes down to the validity metrics of the assay. Also, we see as well that these available platforms, they tend to build on previous experience. They're going from version 1 to version 2 to version 3, and their performance is consistent. And also with that, a lot of knowledge comes about the biospecimens that can be tested, which are, of course, are important when it comes to your study design or experimental plan, as it relates to what biospecimen you're going to use. So the vendors have previous knowledge as well on, how the biospecimen should be processed, how it should be stored, whether freeze thaw affects it. Where you're talking about other omics technologies, we focused on microRNA sequencing. we actually tested that ourselves to make sure that there wasn't any effect. But interestingly, when we looked at microRNA sequencing, sort of considerations again for omics technologies when you're choosing a platform, even if it's a novel platform, is to think of the technical variation and potential systematic biases. And what we found with the microRNA sequencing was that, you know, we actually looked at different kits and different pipelines because the output could be very different. The take-home message there is that when you look at this data, it's probably it's a better idea to compare data from the same process. So again, different processes, different platforms. Metabolomics, I would see as another option for exploring omics technologies. I put in genomics into my cartoon as well on a very speculative note. Recent analyses have been looking at cell-free DNA in circulation. And while the focus of those investigations have been on small molecules for cancer therapy, we are now seeing studies related to biotherapeutics. Now, is there an opportunity for multiple pharmacological molecules there? You are looking at the level of the DNA, but if you use sequencing, there's potential to look at the mutation spectrum as well. Aside from the analytical platforms, we're also interested in natural language programming, and we're looking at pediatric molecular targets using that approach. So there could be an opportunity and space there to utilize existing data to mine unstructured, the literature basically, to potentially identify and prioritize other candidates and link that even with machine learning to look at, you know, unknown relationships. But the problem with that is, or an issue with that is that the algorithm would have to be validated, and that requires empirical evidence as well.
Great. Thank you, Paula. I would like to invite all the panelists to weigh in. So knowing that we have all these futuristic and really nice technologies, what do you think the steps that we can take to broaden and speed up the evaluation of these potential PD biomarkers, and of course, not just the novel technology? Any?
I mean, I think when you look at speeding up and thinking about how to apply these data across the different areas, I mean, one of the ways is to gain clinicians' acceptance and other acceptance across the field as a whole. It's, you know, I applaud the efforts being performed to try to develop and really validate these markers, et cetera. I think the challenge will be getting, you know, expanding it beyond the regulatory sciences and into other areas to gain full acceptance that clinicians will buy into this, or other stakeholders will buy into these markers as valid surrogates for similarity, for no clinically meaningful difference, for those things that payers, clinicians, patients, and others in society want to have a deeper understanding of. So again, it's education, it's validation, and it's kind of approaching it from multiple areas. It's a big ask to do all of that.
Thank you. Bern, I saw your hand raised. Please go ahead.
Yes, thank you, Yame. I think one of the challenges is, of course, that finding potential novel biomarkers that might be useful in biosimilarity assessment is by default a discovery effort. It's not really a development effort. Whereas I think most companies that are in that space are ultimately focused on development. So it's basically a big stretch to make this additional discovery effort without unclear outcome, obviously, because you really don't know whether you find a potential biomarker. So I think what from my perspective, the step to move this forward is really a dialogue, a continuous dialogue between regulatory agencies and industry to ultimately develop a consensus for a set of biomarkers in a specific therapeutic area, or even more specific for a specific drug product. So basically similar to the product-specific guidance documents that you already have established. If a document like that is there, it's basically the blueprint and the roadmap for a company to go ahead and avoid the potential discussion of whether the biomarker is actually something that is useful in that specific drug product or not. I think if that is established, I think the acceptance level may increase, because it really is this uncertainty about whether this is a good approach is taken away, and you don't embark on this famous fishing expedition where you try to find something and you really don't know what you get.
Could I also ask you please to comment on the role of academic institutions in this particular endeavor? Is there a place for the academic institutions to contribute to this work?
You know, I'll take a stab at that. I think it's challenging to see how that partnership would work. I mean, as I hear this and think about it, this feels like very much a regulatory science industry interaction. The challenge is then getting to that place and then going beyond it for development and then publicity of these markers as they go forward. Just to make another quick comment, oncology particularly has suffered from biomarkers, not necessarily benefited. And I think about PSA and CA-125 and CEA and all the tumor markers that had so much promise back in the 80s and 90s, which have been tossed out for markers of anything other than potential disease tracking, but they're certainly not used to get drugs on the market. We still use radiographic data. We still use accepted markers of drug activity. And so biomarkers in some ways really may be a hindrance historically to adoption of this within the academic space and within the clinician space. So that's even a bigger hill to climb in some ways than it might be in other disease areas.
Thank you for that thoughtful historical perspective there. That's wonderful. A lot to think about. So we have a question regarding what when we are using multiple PD biomarkers, how do we assess the PD similarity? Is it what are the criteria? So maybe I can invite Chin to please address this question.
Sure. Yeah, this is a really good question. And as I mentioned in the third case, that case used a combination of both the targeted engagement, the upstream, the biomarker, and a series of downstream signals. So in that case, the requirements sometimes may be a product specific, but usually we will not expect all the potential biomarkers are required as the primary endpoints. Maybe, the target engagement upstream, is one of it, and then choose the most relevant with the strongest data evidence, one of the downstream signals as another primary endpoints. And the additional biomarkers could be included as the secondary endpoint to increase the confidence. Thank you.
Thank you, Chin. And there is another question that's regarding when we have biosimilars for the same reference product that actually maybe the first one that got approved has the comparative clinical study, and how about subsequent one? Can the subsequent biosimilars actually have just PK/PD approach and not a comparative clinical study? I would like to invite Sarah. Would you like to address this question?
Sure, thanks for that question. As I indicated in my introductory remarks, not all within a program, not all programs are exactly the same. And so certainly if and when, an applicant proposes to use a biomarker that we consider to be well justified, then absolutely that would be another approach. So there can be more than one approach to a biosimilar drug development program, whether it's from the clinical program space or otherwise. And you will see potential differences between even programs between products that have more than biosimilar products that have more than one approval to the reference product. You'll see that there might be differences between even the comparative clinical study and or the PK and PD studies that had been conducted, whether it's slight nuances to design, number of subjects or patients that were enrolled, and potential differences in endpoints as well. So that is absolutely an option. Just because a product may have been approved via one clinical program doesn't mean that that is the standard or the expectation for subsequent programs.
Thank you, Sarah. So we're at time, and I really enjoyed this session. Very, very thoughtful discussions. We have a lot to take back and really discuss and evolve our biosimilar evaluation process. Really, thank you very much for your input.
Hello, everyone, and welcome back from our break. My name is David Strauss, and I'm the director of the Division of Applied Regulatory Science in FDA's Office of Clinical Pharmacology. And it's my pleasure to kick off this final session, which I think will be a very exciting and interesting session with a great panel discussion. Mark McClellan will be joining me for the closing remarks following the session. During session 5, we'll hear from 3 speakers and then have time for a panel discussion and questions from the audience. Our presenters include first, Dr. Peter Stein, who's the director of the Office of New Drugs at FDA. And this office does oversee the clinical review of biosimilars. And Dr. Stein will be presenting on regulatory considerations when assessing biosimilar drugs with pharmacodynamic biomarkers. Next, we'll hear from Dr. Jian Wang, who is a division manager overseeing biosimilar review at Health Canada, and he'll be presenting on a Health Canada perspective on using pharmacodynamic markers for biosimilar development and approval. And our third talk will be from Elena Wolf-Holtz, who is chair of the Biosimilar and Medicinal Products Working Party at the European Medicines Agency. Dr. Wolf-Holtz will be presenting on EMA's thinking regarding PD biomarkers and how thoughtfully designed programs can rely on different combinations of study data to support biosimilar approvals. And this will be covering PD biomarkers in clinical pharmacology studies and also in patient studies. Now, Dr. Wolf-Holtz, she actually participated in the panel discussion for session 1 yesterday, but will be giving a pre-recorded presentation in today's session 5. After the 3 talks, we'll be joined by additional panel members. This includes Abhijit Burway, who is Chief Medical Officer at Viatris, Janet Franklin, who is the Executive Medical Director and Global Development Lead for the Global Clinical Development Group at Amgen. Also, we will have Dr. Xiaomei Huang, Deputy Director of the Office of Clinical Pharmacology at the Center for Drug Evaluation and Research at FDA, and Dr. Sunichi Okudara, Deputy Review Director in the Office of Cellular and Tissue-Based Products at the Japan Pharmaceuticals and Medical Devices Agency. So this will be a robust set of talks and discussion. We have representatives from 4 different regulatory agencies around the world, along with industry representatives. First, I'll turn it over to Peter from FDA. Peter?
Thanks, David. And I don't know if my camera is working, but hopefully you can hear me.
Yes, hear you and see you.
Oh, okay. Well, I don't see myself on the screen, but that's just as well. Thanks, David. And first, I'd say that I feel sort of fortunate coming towards the end of this really outstanding workshop, I've fortunately been able to attend a number of the sessions. And what I can say is that a lot of the comments that I'm going to make will be little more than highlighting, I think, some of the really useful points already made by so many of the other presenters. Next slide, please. So I won't go through this in great detail because I think we've talked in this meeting already extensively about what we're about in terms of the definition of biosimilarity. And of course, the distinction that Sarah Yim made in one of the early presentations between the generic framework and the biosimilar framework, when we expect there to be some differences, and we're trying to evaluate the clinical relevance of those differences to draw a conclusion that there's no clinical meaningful differences between the biologic product and the reference product. Next slide. Again, I think in the framework of generics, we believe the chemical can be exactly copied, and in the framework of the biologics, we recognize it can't be exactly copied. Biologics are complex, obviously, mixtures of variants with different post-translational modifications and charge differences and the like. The good news is that we have improving analytic capabilities that give us very detailed assessments, typically using orthogonal approaches in our structural analysis, so that we can identify these and characterize these differences. The evolving understanding of how differences in a particular glycosylation patterns, charge patterns, relate to whether it be PK or functional differences is obviously improving. And that's a very important point in terms of our ability to gather information and to understand the implications of these differences. But of course, across specific drugs, there's varying understanding of how a particular structural difference and any in vivo functional difference relates to the ultimate drug's mechanism of action. Sometimes we understand this exceedingly well, and sometimes we don't have a full understanding of that. And obviously, these are areas where further research is necessary. And we've heard about quite a few of the efforts that are ongoing to try to understand these and correlate structure and function to be able to make good decisions about whether we've reached a conclusion of biosimilarity. Next slide, please. I won't go through this pyramid, which I think you've seen in numerous prior presentations over the last 2 days, but just emphasizes the stepwise approach that we typically take, with the base of that pyramid being the analytic data set that we develop to understand the differences, then characterize those differences. Next slide. Again, this comparative analytic assessment at the base of the pyramid has been improving over the years with new assays, ability to look at things in orthogonal ways that give us more and more information in characterizing differences. And the most important thing is for us to understand what the implications of these small differences might be, whether they imply potentially relevant differences in the mechanism and ultimately in the drug's efficacy or safety, or they would be unlikely to contribute to meaningful differences. Next slide. Now, to turn to pharmacodynamic biomarkers in the pyramid, in a sense, or in the sequence, the stepwise sequence, obviously we start with understanding the structural differences, such as post-translational modifications. And as I said, these are almost always detected. And we may now, with our increasing understanding of the relevance of particular changes, of specific changes, understand whether they suggest that they might lead to functional differences or might not. But this obviously contributes to our residual uncertainty, particularly if we don't understand the potential implications of a particular specific difference between the biosimilar candidate and the reference drug. In vitro functional assays can obviously help us. We've talked about that in this meeting. They may help us understand whether a structural difference seems to have an in vitro functional correlate. And particularly if we understand the mechanism of the drug, that may help us to address residual uncertainty. Otherwise, we go to clinical pharmacology studies, obviously looking at the pharmacokinetics of the drug, but also here where we can look at the pharmacodynamic effects of the drug and exposure-response relationship. And obviously a key is whether we have biomarkers that actually reflect the drug's mechanism of action or mechanisms of action. The extent we have information that supports the role of that particular biomarker in drug response and whether it's on the pathway to the drug's mechanism and its effectiveness in treating the condition that it's targeted to treat. Do we understand all of the mechanistic pathways contributing to the drug response is an important question, but also when we characterize the exposure response, can we see that the exposure response for a pharmacodynamic marker that we would be looking at overlaps? And obviously, if there's residual uncertainty, we move on to comparative clinical studies. Next slide. If you just click through to the whole slide. Now you've seen a version of this slide, and I'll focus on the middle panel just to say that we know that drugs may have multiple different perturbations of biologic systems. The biomarker that we may be looking at may not relate to the clinical outcome, or there may be multiple different biomarkers that we can detect that do in some way reflect the clinical outcome or other biologic responses of the drug, and understanding whether we have the top box with a very clear-cut role of a biomarker or the middle one is something we often don't exactly know. Next slide. Again, the purpose of the PD biomarkers is to evaluate residual uncertainty based upon the base of the pyramid, the analytic information that we have, and with each additional information, the PD biomarker can clearly provide more information. Next slide. Ultimately, our goal might be to be able to make an assessment based upon clinical pharmacology after the analytic information is available. Next slide. I won't spend much time on this slide because we've talked in multiple different presentations about the characteristics and the 5 criteria that we use to look at biomarkers and whether they might have a role in being able to support biosimilarity or contribute to the evidence to support biosimilarity. So I won't go through this further. Next slide. Again, in terms of the uses and value of pharmacodynamic biomarkers, one of the values that we've heard about in several presentations is that they can be less variable, and they can be attained in studies in human volunteers where particularly the variability may be less. But of course, there are certain drugs that can't be studied in healthy volunteers. We also may be able to use lower doses in healthy volunteers where it's on the steep part of the exposure response, which may not be allowable or ethical in the setting of a patient study. If the biomarker is well understood and directly on the pathway from drug exposure to this response, this may be a very strong support for the conclusion of biosimilarity. And we have the example of the glycemic insulin clamp for insulin biosimilars or interchangeables. But even without the biomarker reflecting the primary drug pharmacology, we've also talked about the potential for patterns of biomarkers to be comparable across biosimilar and reference drug, and that help us to be reassured that the drug is biosimilar to the reference drug. Next. The limitations that we not infrequently deal with is that we don't know all of the relevant mechanisms of action. And so we may not have the simple picture that I showed on a few slides ago with a biomarker interposed between the drug administration and the effectiveness response. Now, we heard also that it's not absolutely necessary that it be entirely correlative with the efficacy, but certainly when it is, it makes the contribution of that pharmacodynamic marker potentially more useful and more readily understandable. I also think we have to recognize that there are situations where the efficacy dose response or exposure response and the biomarker exposure response may not correspond. And we've talked about different approaches in that circumstance. Next slide. I do want to just note the limitations of comparative clinical studies. Of course, this is where we end up if we believe there's residual uncertainty that need to be addressed. This is the top of the pyramid, and we need to design this study to be adequately sensitive to detect differences if there are differences. But I think it's also important to recognize that there's variabilities of clinical endpoints and biologic responses that lead to variability. And so these can pose practical issues. I mean, the challenge here is that the residual uncertainty may suggest only a small potential clinical difference, and yet study powering may not be sufficient in a pragmatic way to detect the small differences. So that is a challenge we have to recognize. Next slide. And I think this has been discussed before, but I think it is an important case in point in terms of where we can evolve to as we have better understanding of sort of the structural functional relationship and have experience with particular categories of drugs. We know that in prior status that insulin biosimilars and interchangeable drugs included comparative clinical studies as these drugs were converted from 505(b)(2) to biosimilars. the full package was expected and supporting the assessment of immunogenicity. We had internal discussion, a detailed assessment with multiple different disciplines contributing, recognizing that this is a well-characterized molecule, well-understood mechanism, limited relevance of immunogenicity in general. And we were able to put out a guidance that concluded that in general, a comparative clinical study was not necessary, and that we could conclude biosimilarity in immunogen and interchangeability with more limited information, detailed analytic comparison, and a comparative clinical PK/PD study. So great progress in that particular drug class. And I think as we get to know other drug classes, we'll hope to make progress in the same direction. Next slide. And I want to thank you for your attention. I look forward to the panel discussion.
Okay, thank you very much, Peter. Next, we will hear from Jian Wang from Health Canada. Jian?
Hello, can you hear me?
Yes, loud and clear.
Okay, thank you. So, well, first I want to thank the workshop organizing committee for inviting me to do this presentation to give a Canadian perspective on using pharmaceuticals, on using pharmacodynamic markers for biosimilar development and approval. Next slide, please. So this is kind of a highlight, just going through that, the data requirement for biosimilars and the PD endpoints in biosimilar studies and the Canadian guidance, what we ask for the PD biomarkers, and also challenging, in using novel PD biomarkers. So next slide. So first, let's just give you a brief overview how biosimilars are regulated in Canada. So in Canada, biosimilars are regulated as new drugs, which also include copies of low molecular weight heparin and recombinant DNA peptides. So which is regardless, what's the peptide size, as long as you go through recombinant, that's the technology to use, to make the peptide, the copies of this peptide will be regulated as biosimilars. So this table shows, next slide. So this table shows that given the complex nature of biologics, so as we know, more data are required for biosimilars to demonstrate similarity to their reference products. So in order to support market authorization. So PD study is one set of data that can be used to confirm similarity. Next slide. So in the PD study section, the Canadian 2016 biosimilar guidelines states that in a PD study, so PD markers should be relevant to the mechanism of action of the drug, but may not need to be established surrogates for efficacy. So basically we are talking about PD surrogates and PD biomarkers. Next slide. So also in the PD study section, so which is also, we talk about, how PD markers can be used in clinical efficacy trials. So in this case, a clinical efficacy trial may not always be necessary, right? So we also state that where there is a clinical relevant PD endpoint, in such case, a scientific justification is needed, and the safety as well as comparative immunogenicity data are still required. So there is a certain flexibility, for using PD biomarkers or PD surrogates in the comparative clinical trials as well. Next slide. So one major question of using PD biomarkers or surrogate markers that need to be addressed by all parties is about sensitivity. If it is a PD surrogate, it should be clinically relevant. If it is a PD biomarker, it has to be relevant to the mechanism of action or induce a targeted effect. So a PD marker should be sensitive and correlate to PD changes. I think that has been discussed in yesterday's presentation. Also, we have the cases because most these single dose PK/PD study for biosimilar is conducted in healthy subject. So when we use a therapeutic dose for patients, that may induce a ceiling effect in healthy volunteers. So in terms of PD response, so therefore, most time we recommend the sponsor to use a lower dose, which it can be therapeutic dose, as long as the response curve still within the linear range. Next slide. So this is a table and shows what has been used or what has been in the guidance, and some are proposed in the study. A list of PD endpoints, either considered as PD surrogates or PD biomarkers in PK/PD studies. Next slide. Also, this table shows that the PD endpoints used and proposed in efficacy studies. So, for instance, the pathological complete response or total, PCR or total PCR has been used to authorize, oncology biosimilar product. However, other endpoints, they can, in clinical studies, they are considered as a PD endpoints or PD biomarkers as well, but there may not be well-defined, and it doesn't mean the regulators always agree upon these study endpoints because some are just kind of under investigation. Next slide. So one thing I want to point out, for stakeholders, Health Canada considers that the 95% confidence interval for mean ratio should be within the predefined acceptance limits of 80 to 125%. All 95% confidence interval is within the predefined equivalence margins. So these are the endpoints parameters required by Health Canada, which is not harmonized with all other major regulatory agencies. So this is the one parameter. Actually, when you look at the PK/PD endpoints, they may not always in line, among different regulators. So when sponsor designed a trial, particularly when they look at the PD study endpoints, they have to make sure they meet, the different regulatory requirements, at least for the sample size, for the power calculation. So next slide. So we said these PD endpoints can be used in both PD/PK studies and clinical efficacy studies. So regulatory requirements for biosimilars are evolving. When you look at all these guidance documents, from more than 10 years ago, and compared to requirement for now, you can see that there's a lot of changes. So progress in analytical science and experience accumulated with biosimilars, so continue to change regulatory requirements. So generally speaking, it's leading to reduced requirements for clinical trials. However, in the meantime, so PD endpoints have been used to address residual uncertainties or to be used to detect clinical meaningful differences between a proposed biosimilar and a reference product. I think there is debatable, what would be considered as, meaningful. I think we have to look at a totality approach, not just from PD perspective. I think we had to look at what has been compared on the quality side, functional side, and also the PK/PD altogether. So for products like filgrastim and insulin, many speakers have mentioned that the PD endpoints are well established. For others, they are less refined, right? So as the purpose of using PD endpoints is to confirm similarity, a correlation between the PD endpoints and the clinical outcomes is not a requirement. So therefore, PD biomarkers can be used in the comparative study for marketing authorization of biosimilar. Next slide. So to date, PD biomarkers have not been widely used, right? So, for biosimilar development and approvals. So moving forward, these should be opportunity to use PD biomarkers alongside or in place of comparative clinical trials with efficacy endpoints. So new PD biomarkers need to be identified and explored so that biomarkers data can be used in clinical pharmacology studies. So with this information, for more biosimilars can be authorized without requiring comparative clinical studies to demonstrate similarity based on efficacy endpoints. So for sure, to extend PD biomarkers beyond surrogate endpoints, it will be important to invest in evaluating and synthesizing the available information from the literature, conduct pilot studies, complete model-based assessment, and investigate novel or emerging technologies. So glad to see FDA is doing some pilot studies in this area, and also we have heard that industry is also doing some pilot studies in this area. However, we also heard from others that they need to investigate a lot, in this area. So it may not be that feasible. So we know that, so there are still a lot of challenges in this area using novel PD biomarkers, such as there is lack of historical data. So how much do we know the assay is sensitive? How do we set the equivalence margin, when we design a trial? And how much data needed so we can generalize and grant all indications for biosimilar product? Next slide. So for sure, everyone will benefit from using biosimilars. Regulators in the industry need to work together to ensure that high quality biosimilars reach patients as quickly as possible without compromising the high regulatory and scientific standards. Next slide. Well, I think this is my last one. So thank you for your attention. I'd be happy to participate in discussion and answer questions. Thank you.
Thank you for those remarks, John. And last but not least, we'll hear presentation from Dr. Elena Wolf-Holtz from the European Medicines Agency. This is a pre-recorded presentation, so she won't be able to join this panel discussion. She did join a panel discussion yesterday.
Good morning to everyone. Welcome to the second day of the Duke FDA workshop on pharmacodynamic biomarkers for biosimilar development approval. I'd like to thank the organizers for the opportunity to present our current thinking in Europe at this time. The way I structure the talk is that I want to talk about the status quo in Europe, then maybe give you some insights into what we've learned from the experience gained so far, and then some thoughts on weighing the evidence, that is PK versus PD versus other clinical trial components. This is really a slide, introductory slide to reassure you that Europe is very much committed to optimizing the biosimilar regulatory framework and the registration process. You see here two documents, the European Medicines Agency Network Strategy to 2025 and the EMA Regulatory Science to 2025 document. And in both, it is clear there is a support for patients that they receive timely access to affordable medicines, and biosimilars are mentioned in both documents as being a priority. So far, and that is up to September 16, we've had 116 marketing authorization applications submitted, 103 have been reviewed, and we have 80 positive opinions with 66 substances on, that is products on the market. And you see an overview of these products listed. We have guidelines in place. This is here the overarching clinical guideline that actually refers to PK and PD studies that may replace efficacy studies for approval, and it gives 3 conditions that PD markers should meet. Number 1, there should be a comparable dose response or concentration response relationship. Number 2, for a particular PD marker or biomarker, it is expected that it is an accepted or validated surrogate marker, and equivalence margins should be defined a priori before the trial begins. We also have product-specific biosimilar guidance. We have guidance for both interferons and, of course, for EPO, granulocyte, CSF, and human insulin. We have a specific mentioning of the pharmacodynamic markers that we would like to see. Interferon alpha is an exception in that actually we would expect a fingerprint approach and do not have a clear understanding of the mechanism of action of these components, but this is really the only drug where this would be the case. In all other instances, we have a pretty good understanding of the mechanism of actions and clearly defined measures. New is recombinant somatropin. So far, we were of the opinion that you have to go for a hard clinical endpoint, that is change in height velocity as clinical outcome parameter. But now I think the evidence is building that PD parameter like insulin, like growth factor, IGF-1, could be a preferred PD marker, and the existing guideline is now currently under revision. What have we learned so far from the experience we've gained? We have currently actually from 2010 until the end of 2020, 445 scientific advice letters that have been written with regard to antibodies, antibody fragments, and fusion proteins. We've had 312 requests for 23 substances, which you see listed on the left side. You don't need to read all this right now, but there is a range from rituximab with altogether 48 requests up to now. And just recently also nivolumab with the first requests for this particular PD-L1 inhibitor being developed as a biosimilar. We have discussed PD markers for monoclonal antibodies. You're aware trastuzumab with PCR at surgery, also pertuzumab, PCR at surgery, natalizumab in multiple sclerosis. We would expect MRI, that is as a measure patients without new gadolinium enhancing T1 lesions, but also alpha-4 integrin receptor saturation. I think for eculizumab, it's well accepted to look at the levels of LDH and how they behave in patients. And for denosumab, there is some discussion on the use of two PD markers, one being lumbar spine BMD, that is bone mineral density at 12 months. This is a measure with greater clinical relevance. And on the other hand, a marker like a bone resorption marker, serum C-terminal telepeptide, that is SCTX, which has a clear dose response and a larger dynamic range. These are all published examples that I have presented. There is some discussion as to the relative importance of BMD in postmenopausal osteoporosis women on the one hand, or on the other hand, a bone resorption marker as SCTX. I think there's also a different view in different legislations. Here we in Europe would like to see both actually as co-primary endpoints. And one of the reasons why we are not satisfied with just having the serum CTX is that we think that the strength of the correlation between changes in serum CTX and clinical outcomes is not yet fully established. The question is, of course, if this is something that could be revisited and there could be possibly proposals that applicants come forward with. Whatever the case, I think with all the cases that we do see in the submissions with regard to demands on a PD marker, it may be hard to just have PD markers that already are validated surrogate markers. And it may also be very hard to predefine equivalence margins because the data does not exist readily. We have also looked at differences or omissions in the comparability exercises that did not preclude a positive decision on biosimilarity. You see here 14 cases where there was a, let's say, imperfect file that we reviewed. And it is quite clear that a formally failed clinical trial may still be acceptable even for approval of a biosimilar, as seen with the trastuzumab biosimilars to cases that have been solidly reviewed and with the totality of evidence granted that did not preclude a positive decision. Similarly, also a formally failed PD study may still be acceptable if supporting evidence coming from PK trials and of course a solid quality dossier and the other pieces of the comparability exercise being strongly in favor of such a dossier. On the other hand, clinical efficacy data clearly cannot overrule the failed PK trial. And this is a lesson really to keep in mind. PK trial really seems to be the sine qua non, the most sensitive part of the clinical component of the comparability exercise. So what about weighing the different aspects of a clinical component of the comparability trial, that is an efficacy or safety and immunogenicity trial versus a PD trial versus a PK trial? And here I think we've seen an efficacy or even safety immunogenicity trial may, under certain circumstances that are also laid down in the guidance, be omitted. And even the PD component may be overruled. And as is written in our guidance, the possibility exists of considering PD not a primary endpoint, but rather a secondary endpoint, thus allowing descriptive analyses. However, PK trial, again, always is necessary. What about other examples? Weighing PK versus PD versus efficacy trial evidence. Does a comparability exercise, is that conceivable without a PK study at all? Yes, is conceivable. Just think of intraocular administration of a substance like aflibercept or also ranibizumab. Here it is not possible reasonably to measure PK systemically, and therefore that may be foregone, and rather hard clinical endpoints may be measured in the efficacy trial. Is a PK trial conceivable without PD markers? Yes, of course. I mean, this is what we see for monoclonal antibodies in 18 of the 23 substances that I've just listed for you. Actually, we don't have defined or agreed markers yet. We have proposals. We're thinking of proposals on the table, but it is still a matter of debate. And I've just reviewed with you the case of insulin glargine. What about the other way around? PD study without PK data. Is that conceivable? Yes, even that is conceivable. Just think of heparin. Here PK is not measurable, and here we would rely on PD, at least on factor 10A or factor 2A PD measurements. And then you could think of romiplostim that has unacceptably high PK variability, and here we would rely on PD marker thrombocyte count. We would like to draw your attention really to the tailored scientific advice procedure that is now a new regulatory pathway adopted by CHMP in February of this year. Because really here, if you believe you have a convincing, very strong quality package, then the other components that would constitute a clinical component of the comparability exercise, that is PK, or PD, or a combination of both, or possibly even additional efficacy data, really can be discussed. And before really using, let's say, poor PD markers or insufficiently validated PD markers, really there could be a view that in addition to a convincing quality package, a PK study, which also will gather some safety and immunogenicity data, could possibly suffice. So with this, I come to an end. I hope that this was a helpful insight in the current thinking that we have currently in Europe. And with this, I would like to acknowledge also my colleagues, especially from the Biosimilar Medicines Working Party, but also other working parties at EMA. And thank you for your attention.
Okay, thank you very much. If the Duke Margolis folks can unlock my video from turning on, that would be great. I'd like to open our discussion by turning to our colleague, Shinichi from Japan's Pharmaceutical and Medical Devices Agency for initial remarks. Shinichi, would you like to provide some thoughts about yours and PMDA's approach to PD biomarkers for biosimilars?
Yes, thank you for inviting me to this wonderful session. It's a great honor to take part in the session. So biosimilar guidance in Japan says that PK/PD studies might replace efficacy studies when justified. So where PD biomarkers represents the mechanism of action and or more sensitive than endpoints normally used in clinical efficacy study, and then the PD study can replace efficacy study. And I think this is the case in insulin products or few glycerin products. So I think our basic thoughts are similar to other regulatory agencies. Thank you.
Okay, thank you very much for those comments, and thank you for being with us here in what you can describe as either really late at night or really early in the morning in Japan. So we really appreciate you joining us live. I'd like to go on next and get some thoughts from the industry colleagues that are joining this panel. I'd like each to provide each of them an opportunity to briefly remark on the workshop's themes and regulatory activities that might advance the application of PD biomarkers. And first, I'd like to turn to Janet Franklin from Amgen.
Are you able to hear me now? Unmuted, I hope.
Yes, loud and clear.
Great. Yeah, thank you so much for the invitation to the workshop. I really found the discussions in terms of the accomplishments as well as some of the challenges really enlightening, and also applaud the FDA for the research they're doing in this area. You know, as a drug developer, I think, you know, some of the things that came to mind as I was listening to the presentations and of course in discussions back at Amgen as well, were, you know, really looking at how we've already established our biosimilar programs thus far. And that's been to look at all the components that are needed for biosimilarity data packages to date. And then we realize, of course, there's a whole range of biologics. So I think, you know, understanding that complexity is where we need to have this so-called customization, or this ability to say, this is the requirements for a biosimilar pathway. And then when you get to the issues of PD biomarkers, then the examination on a product by product level, biologic level, what is going to be applicable? Because we know that there's good examples for biologics that have a really well defined pathway for PD biomarkers that are suitable to be used. And then we have certainly another group that for which we don't have any type of confidence yet, not sufficient confidence in terms of basing a whole regulatory development program on that. So I think we're finding that this will shift over time, I would presume, but nevertheless, I think that has to be kind of one of the ask in terms of the guidance that will be developed in the future, because there is that range of complexity and also the range in terms of what we understand about PD biomarkers, you know, for a variety of illnesses, as well as a variety of mechanisms of actions, and some of which are not really well understood. So I think it just enlightens us in terms of the fact that there really has to be some kind of a approach on a, you know, case-by-case basis until we have, you know, more data to say that we have other ways to have the confidence. I was also struck too by a comment was made yesterday by one of the academicians. I'm forgetting the name now, I apologize. They did talk about, you know, the other issue is about the education of clinicians and end users, patients, and others in terms of having that understanding and confidence in terms of the data sets that we're providing to them, and therefore, you know, why the approvals were possible, so to speak. And so I think, you know, having the science behind the use of PD biomarkers will be kind of, you know, utmost in terms of when they're developed and utilized in a program, when they're not. And I would also just put out the ask for the most early and transparent conversations that you can have with our agency colleagues is most helpful, because, you know, again, the work done in the early parts of the program for the, you know, analytical assessments, to meeting all the criteria for the critical quality attributes is really a big part of the program. And so to kind of know early on, where you're heading and what's permissible in terms of perhaps something being different than originally thought, such as not needing the full comparability exercise, or when it's going to be needed anyway. And we also note that many of the drugs that we develop, as many other sponsors, go to various parts of the world and not just to the US. So, we have to kind of weigh that too in terms of, the benefits of the PD biomarker pathway, if it's applicable. And of course, I think right now it's only applicable to a small group versus, of course, the idea of trying to have drugs available to patients throughout the world. So we have to kind of balance those requirements as well from the various agencies.
Thank you very much, Janet. You brought up some great points about customization and harmonization around the world. And I think those will be some good points to return to in the broader discussion after these initial comments. Thank you very much. Next, I'd like to turn to Abhijit Burway from Viatris. And so Abhijit actually gave a brief talk in session 1 yesterday, and it would be good to get Abhijit's reaction to now the workshop overall. And I would note in your talk yesterday, Abhijit, you mentioned how qualified PD biomarkers had been used to really streamline development for a handful of cases, and the challenges remain where we don't have suitable biomarkers. And you commented a little bit about using orthogonal approaches or fingerprinting approaches. And we actually heard that come up in Dr. Elena Wolf-Holtz's presentation from the EMA today about their expectations for a interferon alpha product that would include that. And if you had any comments on that aspect, that would be terrific.
I think, thanks, David. I think, first of all, I'd like to thank both the Duke as well as FDA for this workshop. I think it's extremely timely. And as I sit here and listen to Dr. Stein, I remember the insulin workshop that happened in May of 2019, where we presented a case. And before we know it, I think in November 2019 was the revised guidance as it relates to immunogenicity. So clearly, from a regulatory perspective, I think there is a lot of understanding as well as a movement in terms of making sure that we develop these products, increase access to patients. I think we are now at that crossroad where, I think, as I call it, the low-hanging fruit, we have been able to kind of get that. We have these validated biomarkers, whether it be for EPO or for PEGs or for insulins. I think now the next level really is for products where we don't have this. And when we're talking about these orthogonal approaches, right? I mean, we talk about it at the base of the pyramid. We talk about the CQAs. Everybody talks as if, everybody understands this. And we do. I think over the last 10 years or so, we have learned a lot about how these CQAs really have an impact, whether it be on PK, whether it be on immunogenicity, whether it be on safety and efficacy. And if we build on that, and if you really use some of those techniques as it relates to some binding data, which really could reflect the mechanism of action. And there was a discussion in immuno-oncology this morning. I mean, these are real challenges in terms of how we design some of these studies. So looking at these things in terms of, we may not fit that 5-point criteria that we talk about, it has to be fitting on those were good for when we knew these biomarkers, right? I think for these things where we can say, can I take 2 from here and 3 from there, right? And that's really what, we wanted to have that discussion. And there was, one of the FDA colleagues did mention about some kind of a composite biomarkers. And I think that is really where we need to slowly move towards, at least have that path for products where we may not satisfy that 5-point criteria, because I think that 5-point criteria is good, but I think we need to build on that solid, foundation on analytical similarity and see if we can take that to that next level.
Okay, thank you very much for those comments. Finally, before getting into the more general discussion, I'd like to invite my colleague, Dr. Xiaomei Huang, to offer additional perspective. Dr. Xiaomei's from Dr. Xiaomei Huang's from FDA.
Thanks, Dave. Can you hear me?
Yes, I can.
Okay, great. I think over the past 2 days, we have heard great feedback from many included speakers, panelists, and also other participants, also the moderators, about the use and challenges of PD biomarkers in development and regulatory review of biosimilars. So yesterday, at the opening remark, Dr. Woodcock mentioned that the PD biomarkers can be used to address the remaining or the residual uncertainty, and can be used in lieu of comparative clinical trial efficacy trials. So David, you yourself, Yo-Ming, Jeff, and others had spoken yesterday, and again, stressed this afternoon by Peter, that the goal of the biosimilar evaluation is not to establish the safety and effectiveness, but to compare biosimilarity of the proposed product with the original reference product. So you also stress that PD biomarker evaluation can be part of a clinical pharmacology PK/PD study, and the biomarkers that reflect mechanisms of action do not need to reflect clinical efficacy or tie to efficacy endpoint. And I believe the so-called validated PD biomarkers that have met the 5 criteria for PD biomarkers that have been discussed by at least 4 speakers in the past 2 days, such as absolute neutrophil counts, hemoglobin, as described in the FDA guidance about how to meet those criteria. I believe they have streamlined the biosimilar development for some products. However, the hurdles exist for more complex biologic products. So yesterday we have heard some of the challenges of using PD biomarkers. For example, there were discussions about insufficient data in the literature, and also some details are lacking about how to set the cutoff points, low dynamic range, the lack of sensitivity. And we heard today on Oncology about IL-18 on study design. And also there was a mention about if we ask for a pilot study, and then there's a definitive clinical pharmacology PK/PD study. And that is maybe more time consuming and not as straightforward as a simple, simple comparative clinical efficacy trials. So, and over the past 2 days, my colleagues have shared some research efforts to define some of the evidentiary framework. So for example, Jeff has shared our pilot clinical study to show, to fill the knowledge gap and then to inform this study design. For example, do we need to do baseline correction? What is the length of sample collection, etc., and to evaluate novel methodologies. So for example, Paula this morning has shared her research data about the potential use of proteomics, the small RNA transcriptomics to find new PD biomarkers. And then Ching has further shared a recent submission, which we couldn't name, but where multiple biomarkers have been evaluated. And so the new tools and methods are helping us moving forward to address some of the issues of PD biomarkers. And finally, I want to mention that Sarah indicated that we're only 10 years out in this BP CIA, and we are still in the teenage stage in understanding the biosimilar development. So with the FDA research, and we hope that we are starting to address some of the knowledge gaps and to inform best practices. We also need to consider communication as discussed yesterday and today. For example, the previous speaker talked about the disappointing CA-125, PSA. I guess we need to educate ourselves. What are these biomarkers used for? The context of use. So in the best guidance, FDA had laid out whether certain biomarkers are used for certain purposes. So I think for PD biomarkers used in biosimilar has its unique place. And I think if we continue to communicate among the regulators with industry, academia, healthcare providers, it will help us to understand whether PD biomarkers a role in biosimilar development. And I think this workshop is one of the wonderful ways to get us together and to discuss the issues in the open and help FDA understand the issues in all stakeholders' mind. Thanks, David.
Yeah, thank you, Xiaomei. Great summary and touching on some of the different talks we heard over the past 2 days. So we've gone through, we've heard the talks, and we've gone through the additional panelists' initial perspectives. I'd like to go back to the first speaker, Dr. Peter Stein from FDA. And Peter, I was wondering if you could provide a little comment, and now that you've heard some of the talks from other regulators on your perspective on evaluating the suitability of a PD biomarker approach that would be in lieu of a clinical comparability efficacy study. And I think there, may in the presentations from FDA earlier in this workshop, there was a lot of focus on, you don't need to have a surrogate endpoint biomarker, but at the same time, I thought it was nicely summarized by Dr. Elena Wolf-Holtz from EMA that you can have a accepted, validated marker. You can have a pharmacologically principled biomarker, and also the points have come up about the questions have come up. Do you need to meet all 5 of these principles or these key principles of biomarkers that we've talked about in the workshop and are in FDA's guidance? And it's not necessarily a 100% thing. It's a weight of evidence. So I was wondering if you could comment on your views on these aspects.
Sure, I'd be happy to. And I thought the presentations from the other regulators on the panel were extremely helpful. But I guess I was particularly struck by the fact that I think there's a lot more overlap and to a certain extent consensus and agreement, at least. And I think there's this disagreement or dissonance in kind of our perspectives here. And I think it reflects sort of an evolving landscape as we understand more about biosimilars and about what we can learn from the analytic assessments and how we might use pharmacodynamic biomarkers. Let me just touch on sort of the last point that you raised, and then I'll come back to the earlier point. Does it have to satisfy all of the 5 criteria? And I think we could all agree that these are I think important characteristics that help us understand the robustness of a pharmacodynamic marker, not necessarily sort of pass-fail requirements. On the other hand, there are certain ones of them that have obviously sort of evident validity and importance. If we have an analytic validation of the assay that suggests it's poor, that isn't going to be a terribly reliable pharmacodynamic biomarker. On the other hand, there may be aspects that that are not as critical. Certainly, as for any assay, if the dynamic range is very narrow, picking out a drug effect and comparing a drug effect from one candidate to the reference drug is going to be very challenging. So I think you build based upon thinking about these criteria. Do you have to have exactly the same time course? No, of course that may not be the case, but we at least have to understand the time course. Where on the exposure response is it? What do we understand relative to its mechanism? I think these kinds of criteria help us evaluate the biomarker, but aren't those that I would say, well, gee, it doesn't meet this one, so okay, we're going to kick it out. We're going to kick it out. I think we have to think really and step back and figure out, like, what are we actually trying to figure out with these markers? I mean, what's the question we're asking, and do they answer the question? The simplest question is where we have a PD biomarker that's directly on the pathway between the drug exposure and the efficacy response. And when we see that, of course, we can all jump up and down and say that's what we are looking for, and assuming that it can be measured accurately and the trials are readily feasible, then we really can make a great story that when we are comparing the drug to its reference product, we can be very confident. That's really where we are with insulin. Patrick Archdeacon talked about the glycemic insulin clamp, and that was some of the earliest research I did when I was an endocrine fellow, so I'm very familiar with that, and it's a very precise, it's challenging, but it's a very precise assay. And so that's the one side of it. But on the other hand, if we have, as your group has been doing, trying to characterize various kinds of pharmacodynamic markers, what are we trying to do there? Well, some of them we might ultimately link to the end product, which is the efficacy of the drug. For others, it just may provide kind of a characterization of the various pathways that are stimulated and or inhibited by this drug. And that might give us insight into the lack or presence of potential structurally relevant differences in the biosimilar to the reference product. If there are multiple different pathways engaged and every one of them has general similarity to the reference product, we're certainly going to be a lot more confident that we're not seeing structural differences that are likely to be meaningful, since all of the engaged pathways appear to be similar, even if we're not exactly sure that pathway A and B are the ones that directly contact or directly connect to the effect of the drug. So my point, I guess, would be that the way I see this, and I really think this is a theme that I heard from our AMA colleague, is that we're trying to sort of build a case that there's no meaningful structural differences, functional differences, and therefore expect no effectiveness or safety differences or immunogenicity differences. And I think as we learn more about these pharmacodynamic markers within each particular candidate, we'll be able to better understand their role and decide whether we can draw conclusions of biosimilarity on the basis of these PD markers, along, of course, with PK and the analytic base of the pyramid, or whether that leaves us with residual uncertainty and we need clinical comparative studies.
Okay, thank you very much, Peter. That was very helpful to hear your insights there. I'd like to we can get some alternating perspectives from regulators and industry members of the panel. Abhijit, I was wondering if you could comment on some of the same points in what you would think about in suitability of a PD biomarker and the sort of, I heard throughout the talks and then through the discussions at this meeting, need for a practical pathway for using biosimilars. And I was wondering if you could share additional thoughts of what you would do now and what would help in being able to use biomarker pathways in a practical setting.
So I think, thanks. I think, David, I think yesterday we briefly touched upon it, which is really this development conundrum, right? Because, this is not like a novel product development. We are developing a biosimilar. It's race to be first, right? It's almost like a generic product to a large extent. And we do that a lot. And honestly, the regulators' product-specific guidance has really helped us. So I think one of the first things is that if regulators can come up early on in terms of a product-specific guidance, as we have done with insulin, I think it can be of incredible help. Now, we're still learning, but I think that is definitely one area which is important. And I think that is what we need to think about, that if there is no agreement with the agency, or if these are exploratory in nature, then the default for us would be to just go ahead and load a study, efficacy study, and we can talk about it. I think so that would be really helpful if we are able to get product-specific guidance early enough, at least some thoughts, so that two things, right? One is that we have some guidance, number one, and number two, it becomes some more of a level playing field, right? So that it doesn't mean that, you've given feedback to one sponsor and then, other people are kind of grappling in terms of it or trying to reinvent the wheel. So yeah, there are pros and cons of that approach, but I think this has incredibly helped the generic development of products, and we are seeing all the successes that are associated with that. So that would be one kind of an ask. I think the second ask really is what we also talked about briefly yesterday. And this really, what does that efficacy study look like? Is it sensitive enough? And there are so many efficacy studies that have been done till now over the last 10 years. How many of them have failed and why they have failed, right? If you're able to do that, and that in my mind would be absolutely fantastic, because yesterday Dr. Holmes did mention that they have done a similar approach for immunogenicity. And they say that we don't need to look at immunogenicity. We know that it's caused primarily because of certain process-related impurities most of the times. And if you're able to control the base of the pyramid, then you will not see that. So they have come to that conclusion based on the analysis of multiple clinical studies. I think if that kind of an approach is done to really look at it slightly differently, right? It's like looking at the problem in a different way, where we can say, what is these efficacy studies, these 600, 800 patient studies? Some of them are going to be, as I said yesterday, going to be incredibly difficult. We don't have that meta-analysis of 8 studies to do a robust meta-analysis. So we are going to be doing into that guessing game of that equivalence margin. So is that powerful enough, or is that going to really add value? So I think those will be 2 things that can really help the developers come up, product-specific guidance, and then looking at a retrospective analysis of maybe 100 studies that have been looked at and efficacy, and how many of them failed and why.
Thank you very much. That is, I think, some very insightful remarks, and that theme you just mentioned came out from a few different speakers at the workshop. And someone mentioned, thinking about, I think it was Patrick Archdeacon from FDA, of using a kind of Bayesian type approach of, what is the probability this proposed biosimilar is different from the reference, and our new test, whether it's a PD biomarker, a PK clinical test, or PD biomarker test, or clinical outcome study, how might that impact the result? And I think there's more room for sort of regulatory science development in that area of evaluating the value of these different studies and how we pull them together. I'd like to go back to Dr. Jian Wang from Health Canada and ask if you could provide any additional perspective and thoughts, having heard comments from other regulators and also from industry members and academic and practitioners on, evaluating the acceptability of biomarker approaches, and reflecting on what you've heard from those additional workshop participants.
Yeah, so I have heard a lot of agreement about using the the PD biomarkers or PD surrogates, for biosimilars, development and authorization. I think when we think about the similarity issue and using these endpoint, the PK/PD endpoint, it should not be isolated from, the whole assessment, because I think we base on what we understand from a quality functionality comparison. So if we have the data highly similar from that perspective, so then we'll be add value from PK/PD studies. And also, I think there is still, if there is any disagreement, it's going to be the type of product, right? So which type of product you can use PD endpoints to support regulatory authorization, and which one that there is still not sufficient evidence, or the endpoint may not be sensitive enough. So I think the regulators and the industry should, come together to have some broader discussion, which area we can reach agreement. Simply, definitely those are simple proteins and peptide, it's much easier to use PK/PD data to support. So then for the complex proteins, then the mechanism of action may not be that clear. then there is no PK/PD, markers available. I mean, particularly for the PD biomarkers may not be readily available. So then we need a lot of, further studies. So there is something we can agree upon, based on the analytical, technology and regulatory experience, so that we have gained to reach some consensus. So then we leave other, products need to be further, the relevant PD biomarkers need to be further explored. Thank you.
Thank you very much, Jian. It's really been a great discussion here and a great workshop. I'm going to give a few summary observations and then turn it over to Mark from Duke Margolis to give some final perspective and remarks. I think throughout the workshop, and I highlighted in the talk I gave yesterday some key points that we really focused on in this workshop, is that the context of use for pharmacodynamic biomarkers for biosimilars is different from using biomarkers in new drug development. The biomarkers do not need to be surrogate endpoints for clinical efficacy outcomes. You can use biomarkers based on pharmacologic principles. You could have a single clinical pharmacology study where you assess PK and PD and obviate the need for comparative clinical efficacy studies. We've heard from industry colleagues, we need practical regulatory certainty, also themes of harmonization among regulators is quite important. I think that bringing together the 4 different regulators in this workshop, in this session, is a good start. And as Peter Stein mentioned, there really is a lot of harmonization among the different regulators, but we can continue this discussion. Yeah, I think this has been productive in hearing feedback from multiple stakeholders, how we can improve the efficiency of biosimilar development and enable more biosimilars to come to market and be available for patients. With that, I'd like to turn it over to Mark, and it'd be great to hear any final summary observations and takeaways you had from the workshop, Mark.
Well, thanks, David. First, for all your efforts to bring this together, and second, for facilitating a great session that actually included a number of summary comments and suggestions and thoughts on where we are and where we're headed. I agree with your comments. Pharmacodynamic biomarkers for biosimilar development approval are an important progressing field to help us move towards interchangeability and broad acceptance and use of biosimilars. And there's certainly a number of opportunities we heard about over the last 2 days to help accelerate this process. Really want to congratulate FDA for that foundational guidance with the 5 key principles. That's clearly had an impact and is a good reference point for the further steps that we've talked about over the last couple of days, which has emphasized the iterative nature of progress here, but also the opportunities for accelerating that progress through engagement and data sharing with FDA, and especially, I think, through multi-stakeholder forums like this one that bring together industry experts, regulatory leaders, including from multiple different regulatory agencies around the world, and industry, patient, and other perspectives. So we are going to need more research to understand PD biomarker properties and additional product areas. Some of this may be challenging. We heard from a number of speakers about potential areas, and really encourage building on this workshop with continued FDA engagement. It's been great to see that, and continued efforts to really move forward this shared learning, which I think is very pre-competitive. The applied research support from FDA through the Biosimilars Action Plan to date has been important. I think saw some opportunities here to specific examples, suggestions, cases for building on that in areas like immuno-oncology came up, for example. So a lot to build on from today, and all keeping in mind this ultimate goal of increasing confidence and effective use of lower cost but equally effective biosimilars, and communicating the state of the evidence on biosimilars, not just among us, but to healthcare providers, to the broader public. So this is not just about PD biomarkers, it's a broader process, but this is certainly a key element. So David, I want to thank you and your colleagues on this panel for not only a good discussion on the panel, but also for helping with these insights and next steps. And I'm not sure if you've got any other last words you'd like to get in before we wrap up.
Well, I would like to first add thanks to Duke Margolis. The team there was excellent in working with us to put this workshop on. So thank you, Mark, and all of the Duke team. It was really great to work together on this. And thank you to extensive team at FDA who's contributed to this workshop, and then all of our external partners and stakeholders, the other regulatory agencies, industry, academic, clinicians. And one thing I took away that I thought was quite interesting that I didn't mention before was some of the perspective even about nomenclature of biomarkers, and clinicians will not have familiarity with biomarkers, but actually they do. It's just that we're not calling them biomarkers in clinical practice. And we do really need to think about the nomenclature and the messaging as we advance things forward in this area.
It's a great point, and I'd like to add to your thanks for all the speakers, all the participants, and maybe just one more minute, a few call outs. It was great working with the FDA team on this, including you, David, Yaoming Wang, Jeff Florian, Sophie Shubo, Cheen Soon, Bernadette Johnson-Williams, Tracy Lee, Kristen Prentice, great team. On the Duke Margolis side, Nicholas Harrison, Valerie Parker, Aaron Sewell, Thomas Rhodes, Ethan Chupp, and Morgan Romine all contributed to making this a success. So really appreciate the opportunity to work together, really appreciate the strong and constructive interest in this topic, and I hope all of you have a good rest of the day.